हेलो। हाँ, अरे क्लास के आके भी करूँगा। ठीक है।
अच्छा, ये यहाँ पे वहाँ पे दोनों दिखेगा?
नहीं नहीं, इधर दिखेगा।
इधर दिखेगा स्टूडेंट्स?
वो तो इसमें स्टूडेंट आपको देखेंगे।
स्टूडेंट्स का स्लाइड दिखेगा?
हाँ जी।
ठीक है। उस दिन फिर क्या हो रहा था यहाँ
सर
पे?
वो प्रोजेक्टर में लगाया था उस दिन तो।
अच्छा, वो तो नहीं चाहिए ना।
नहीं।
स्टूडेंट्स ज्वाइन किया है?
हाँ जी।
फर्स्ट दोनों को खोल दीजिए और नीचे वाले कितना ज्वाइन हो गया?
पहले कौन सा वाला खोलना सर?
हाँ, ये दोनों।
ये दोनों सर।
ये वाला पहले खोल दीजिए।
खुद ही चालू कर दे। 2 बज गए।
एक्सेलेंट। फर्स्ट स्लाइड, ये फर्स्ट स्लाइड है। ठीक है। अच्छा, अभी चालू नहीं हुआ। दिख रहा? हेलो, गुड आफ्टरनून।
Good afternoon, sir.
वंडरफुल।
Good afternoon.
गुड आफ्टरनून।
Good afternoon, sir.
We just wait for 1 minute. Let few of your friends to join.
अभी सर स्टूडेंट है 1617107।
इतना था क्या? लास्ट टाइम इतना था।
सर आ जाएंगे 125 के करीब आ जाएंगे।
अच्छा, स्लाइड्स दिख रहे हैं, मुझे नहीं दिख रहा।
नहीं नहीं, स्लाइड देखिएगा सर।
ऐसा?
चालू है।
अच्छा, उनको म्यूट करने का क्या तरीका है? स्टूडेंट नहीं, ये तो मैं भी म्यूट हो जाऊंगा ना।
नहीं, मैंने उनको म्यूट किया हुआ है।
फिर तो आवाज आ रहा हूँ।
ऑलरेडी मैंने उनको म्यूट किया हुआ है।
म्यूट किया फिर वो कैसे बोल रहा है?
नहीं, मतलब अगर ये क्वेश्चन आंसर करेंगे तो वो अपने उससे
अनम्यूट कर लेगा।
ये सब है ना, जैसे मैंने आपको म्यूट किया।
अच्छा,
तो उनको क्वेश्चन आंसर करना है तो उसको दबा के।
फिर बैकग्राउंड साउंड कहाँ से आ रहा है? क्योंकि एक आवाज़ आ रहा है ना बैकग्राउंड में पीछे से।
ये दोनों ना।
अच्छा अच्छा, ओके, ठीक है। ओके, वंडरफुल। सो for the last class, do you have any questions, queries, any suggestions, any criticisms, anything you want to add on to me?
Hello, sir.
Yes, please.
In the last class, you said that the cytoplasm was reducing.
Yeah, reducing environment, yes.
And also that the mitochondria was reducing.
Mitochondria is the most reducing environment, and then the cytoplasm, and then a few of the other organelles like nucleus and all these things.
Yes, sir. So, but in mitochondria, Krebs cycle and all the reactions takes place in which glucose gets oxidized.
Yeah, we say this and oxidized, but think about it, how many reducing equivalents are produced in the form of NADH, number one. And number two, also from the chemistry point of view, just imagine that, if the electron transport chain, electrons has to move, and if there is an oxidized environment, then that is not going to support this entire reaction. And therefore, we need a little bit of reducing condition. At least the reducing environment should be sufficient that the entire reactions of the transfer of the electrons. So when someone is going to kind of cover the electron transport chain, oxidative phosphorylation, you will appreciate that the reducing equivalents which has been produced from the TCA cycle in the form of NADH and FAD, they get funneled into the four complexes of mitochondria, 1, 2, 3, 4, and eventually it causes the transfer of this energy of these electrons and captured in the form of an ATP. That's the currency of the cell. So the entire process is efficient only in a reducing environment, and that is the reason why we need to maintain a reducing environment in the cytoplasm as well as in the mitochondria. However, having said that, there are a few organelles where a little more acidic or oxidizing environment is important. Say for instance, those places where we need to form sulfur disulfide linkages. Okay, in those conditions we need a little more of the oxidizing environment than reducing, because if it is a reducing, then SH will remain as SH. It will not donate its hydrogen to form SS double bond, a single bond. So for kind of fostering the need for the different kind of chemical reactions, we need different cellular environment.
Okay, sir. Thank you.
Sure, thank you. Anyone has any other questions? Okay. It's terrible to start after lunch. There are a couple of reasons. I mean, after lunch, we all feel very lethargic, very sleepy, kind of tiredness and all this kind of things. So it's a real challenge for both of us. I mean, for you guys and for me to, kind of continue a class. Because all of the blood flow is actually through the mesenteric arteries now, superior, interior mesenteric arteries and the celiac arteries, and very little is going through the carotid arteries in the brain. So we kind of feel a little lethargic after the lunch, and it is hard to concentrate. So please take an extra step to keep yourself awake and listen to this important class. Okay. So, shall we begin with the today's class on the structure function of the biomembrane? Anyone can unmute and give me a feedback that the slides are visible and I am audible?
Yes, sir. Yes, sir. You're audible.
Okay.
Yes, sir.
And the slides are visible too, right?
Yes, sir.
Okay.
Yes, sir. Your slides are quite visible.
Okay, so we first begin. So today's class is a little extensive class in terms of 2 hours, but I'm going to give you enough room for kind of stretching yourselves and oxygenating yourselves so that you don't feel very lethargic or boring. But nevertheless, I mean, because of this year, we are kind of squeezing everything kind of a little fast forward. So okay, so today's class is membrane function. So membrane function means biological membranes, obviously, right? And what is their function? So this is something which I kind of written down in the form of a few bullet points. The primary function of the membrane is protection. Obviously, membranes are actually kind of the door of the house. So if the membranes are not there, every cellular contents is going to leak out of the cells. So protection is the primary requirement, the physical barrier. The next thing is that communication. So membranes are very well communicating. See, all the receptors which has been important for carrying the signals, like glucagon receptors, insulin receptors, growth factor receptors, cytokine receptors, anyone wants to add? Anyone is asking anything? Any questions? Okay, I'm continuing. Okay, so the communication is actually very vital, because what happened is that membrane is the hub of communication, it's the platform where all communication takes place. So that's another very critical function of the membrane. You are going to see the detail about this in the further slides. The third important function of the membrane is selectively allows substances in. Again, very important. A membrane is not something where the door is always open and then anyone can go in and anyone can go out. It allows only very few selected substances to come in or come out, and that is the reason why you say it is selectively permeable, or selectively allowing substances to come in or go out. The next is response to the environment. It is kind of a little extension of the communication. So if the membranes are communicating, this means they are able to respond to the environment, external environment. The external environment is not in the control of the cell, right? It is in the control of the entire the entire system. So the systemic control is not within the purview of the membranes, but what the membrane can do is that it can adapt itself so that it can still survive in the face of the fluctuating environment. Suppose you go outside in a Delhi, now the temperature is pretty low, so you go outside and then you get exposed to a low temperature. So how does the membrane respond, versus if someone in summer is, suppose in Jaipur or Rajasthan or even Delhi, where the temperature goes as high as like 45-50 degrees, how the cells are going to respond to these fluctuating environmental conditions? That is another thing which is very important for the membrane. And recognitions, again, it is a similar kind of thing, cell to cell communications, cell to matrix communications, and so forth. We are going to see a little more detail. This is the bullet points just to kind of stimulate you that these are the different functions of the membrane. This is how a cell actually looks like. Exactly, once you take a cross section of this one, the blue is the nuclei, and then this is something which you have already discussed. The mitochondria are actually this one. Over here are the mitochondria. This is the endomembranous system, the endoplasmic reticulum, the Golgi complexes, the cis side, the trans side, and these are the different organelles over there, so lysosomes and peroxisomes and so forth. And then this is the plasma membrane. Now you could see that the entire endomembranous stuff has been pretty much extensive over here, and this is what is actually happening in this entire cell membrane. We are just actually looking outside from an outside. Okay. Then let me shift to the next slide and see what's there. Okay, this is just an image which shows the biological membrane, but what is more important, what I'm actually pulling up over here is that, could you see the lower panel? That's the electron microscopy image of the membranes. You always say that membrane is bilipid layer or lipid bilayer, and this is exactly what you could actually appreciate in an electron microscopy image, where you could see distinctly the two layers of the membrane, the outer layer and the inner layer, and this is the intermembranous space, and that's where the cytoplasm starts with, and all other organelles within this. This is about 35 to 50 angstrom thick. That's how the biological membrane is really, and that's what you could actually see under electron microscope. Now, a few minutes back I was telling that the membrane has to maintain the privacy for the membrane function to execute properly. This is further been given an example in this image. If you see over here, this is the external environment and this is the internal environment. This is the within the cell. Within the cell, if you measure, the sodium concentration is just 10 millimolar. The potassium concentration is as high as 140 millimolar, and the chloride concentration is just again 4 millimolar. But look at the outside concentration. The sodium is way higher, 145. The potassium is way lower, and the chloride is again higher. So what you see is that between the inside and the outside of the membrane, there is a dramatic difference in the ionic concentration. Now think about the situation if the membrane is not selectively permeable, if the membrane is allowing every damn thing to enter within it, this concentration gradient would never have been there. There would have been sodium inside and outside exactly the same, potassium same, chloride same, every single ion same. And once this happens, we say that this is in state of equilibrium, and this is as good as saying that the cell is dead. So whenever you reach a state of equilibrium, that is the state of death, because only on death the equilibrium achieves. Before that, if there is an asymmetric distribution of ions, proteins, lipids, anything, that's the whole philosophy of biology, is that if you see an asymmetric distribution, you should immediately understand that there is an activity going on, there is an active force over there. The cell is living, the organism is living. But once you see that there is a symmetry and there is an equilibration and everything inside and outside is the same, that's the last stage when the cell is possibly undergoing death. The organism is possibly going to die. So the life is basically maintaining this asymmetry, whereas nature will try to maintain symmetry. So that's a very wonderful philosophy between nature and life, is that in nature, according to the second law of thermodynamics, the nature always wants to bring everything in the state of maximum randomness, or in the state of an ultimate symmetry. But at the same time, if you see the cell, what it tries to do is that it tries to maintain and break that symmetry and maintain an asymmetric stage. And this is exactly has been seen over here, that the ionic concentration is asymmetrically distributed. And because of this asymmetric distribution of the ions, we have a potential gradient which has been created. And people are going to talk about hours together that this potential gradient is the basic fundamental point for generating the action potentials, which people in physiology, that's their bread and butter. They kind of study these action potentials day out and day in. So what is the origin of that? The origin of that is the asymmetric distribution of the ions. And why it is that? Because of these membranes are actually allowing a few substances to pass and not everything to go through it. So biological membranes' main function is to segregate the chemicals. That's what is actually been written in the box. Segregate the chemicals means maintain a stage of asymmetry, unequal distributions of the ions. That is very important. Okay. So overview, coming to this overview, cell membrane separates living cells from the non-living surroundings. There is a thin barrier, controls traffic in and traffic out very selectively. That's what we always say, that it is selectively permeable. It allows only some substance to cross through it and more, not all of them. Yes, anyone wants to add anything, wants to ask any question? Okay. And allows, so once I am done with a couple of slides, I'll actually pause and then, kind of ask you that if there is any interactive one, anything wants to add, anyone wants of you has any doubts, so I'm going to go, just give me a couple of minutes. I'm going to finish a couple of slides and then I'm going to pause and then hear back from you, your feedback. Okay. So it's selectively permeable, it allows some substance to cross. Again, you see that the biological membranes, I'm going to come in a little while, is made up of lipids. It's known as lipid bilayer. That's the whole fundamental thing about the membrane. Now when you say lipids are all non-polar, right? That's what we understand by basic chemistry, is that lipids are always non-polar. Or at the most, they will have the polarity, but they will be amphipathic. They will have polarity as well as non-polarity, but the ratio of the polarity to non-polarity is more in the favor of non-polarity. Okay. So if a lipid bilayer membrane is non-polar, it is very conceivable from this information that possibly the lipid soluble organic substances are going to pass through the membrane a little more easily as compared to the polar substances or watery kind of substances which will be prevented by the membrane, because membranes are polar, apolar, membranes are hydrophobic, so they will allow hydrophobic substances to pass through them little easily as compared to the hydrophilic substances. Now this is again not a very universal truth, because again it does not mean that any non-polar substance can pass through it, and again it does not mean that all the polar substances will be stopped either. But then that's what the philosophy of this semi-permeable membrane, that case by case, molecule by molecule, membrane takes a decision what to allow and what not to allow. Okay, we are going to go through that one again. So hydrophobicity versus hydrophilicity, that's what I just mentioned. Made up of phospholipids. Again, I told you that membranes are phospholipids, lipid rich. They also contain proteins and other molecules. And this is how exactly the membrane looks like. Over here, these are the lipid bilayers. These are known as the red ball-like structures. These are known as the polar heads. And the tails over there, these are known as the aliphatic chains of the fatty acids. So polar head, aliphatic chains of the fatty acids. Aliphatic chains of the fatty acids, polar head. That's what the membrane lipid bilayer is. This is protruding outside. This is protruding towards the cytoplasm, and this is the sandwich of this membrane. That's how the membrane looks like in 3 dimensions, with polar heads popping on either sides, cytoplasm and extracytoplasm, and lipid fatty acid chains, which are non-polar in its nature, have been hidden from the polar environment and been sandwiched in between. Within this complex architecture of the membrane, we have these membrane proteins. These are known as the integral membrane proteins and peripheral membrane proteins. We are going to come to that a little while. in just in a couple of slides, but this is how the membrane looks like. It's an ocean of lipids, and embedded within them are the islands of the protein. Again, let me repeat it. Membranes are the ocean of lipids, and embedded within them are the islands of the protein. Okay, that's a very important statement. Don't forget it. This is just a complex structure of the membrane, different kinds of membranes, different substances attached with it. Remember the last day I was talking about carbohydrates playing a very crucial role in giving a lot of function to the membrane proteins. That's what exactly has been shown over here. So this is a picture that kind of summarizes everything whatever you have discussed so far, plus what you have actually spoke in the last class. Okay, so that's what the chemical composition of the membranes. Nothing brilliant in knowing this one, and so don't take an effort to kind of memorize it. Either way you will have this information. This source is actually given over here, so you can always go back and look at it. So I'm just skipping this slide and see what's the next of this one. Okay, I'm going to come to the next a little while. Okay, so these are the phospholipids. So membrane contains lipids, and these are actually phospholipids. Phospholipids, from the name suggests, is that the lipids which has an phospho groups attached with them. This is the one that actually gives the membrane polarity. As I said, membrane is not completely apolar, it's not completely polar either. It's an amphipathic. It has the polar as well as the non-polar groups, whereas the non-polarity is more. But the polar groups is because of this presence of this phosphate residues. Phosphate is PO43. So obviously it has an oxygen, and 3 means minus charge, and therefore it is in polar, right? So that's the reason why you say that this is the polar heads of the lipids. Okay. Now, in a little back, we are talking about peripheral and integral membrane proteins. So let me just introduce that one real quick. Most of you are pretty aware of this one. I'm just kind of summarizing what you already know. Peripheral membrane proteins are the one which are very superficially attached to the membrane. And then we have the integral membrane proteins, which are the one which are integrated part of this membrane. Like you are going to be a part of the AIIMS, you are integrated part of the AIIMS community. So you are an integral membrane protein, if you kind of do an analogy of that. And what are peripheral proteins? Peripheral proteins means the people who come to the AIIMS and do a short fellowship, say for a couple of months, summer fellowships, couple of months, and go. So they are never part of this, they are not never the integral part of this AIIMS community. So think them as an analogy of peripheral proteins. So peripherals are superficially attached, integrals are integrated part of the membrane. If they are part of the integrated membrane, they has to be going all the way through the membrane. Obviously, that's the logic, right? How you could integrate within if you are not a part within the deep core of the system. And that's exactly is the integral membrane proteins that you could see that they have actually been passing all the through the membranes. And there are multiple types of there where they could pass one time or they could pass multiple times depending upon what type of proteins are this. We are not going into too much detail about that. We are going to cover the rest of the other things. And these peripheral proteins are the ones which are superficially attached to the membrane. Now from the superficial attachment, it actually comes down to this particular point that extracting and treating the membrane with high salt or alkaline pH is sufficient to take out these proteins, because these are superficially attached. So if you just add a salt or if you add a little bit of detergent, then you could extract easily the integral membrane proteins. But if you want to do an extraction of the peripheral membrane proteins, a very little force, a very little chemical treatment is sufficient to extract the peripheral proteins. But integral membrane proteins, they are very deeply integrated, and therefore they need a strong detergent, very strong chemicals like urea and other kind of components to extract them. So just by the ease or just by the hard or difficulty with which you could actually extract the proteins from the membrane, they kind of give you an influence or kind of give you a feeling whether this is an peripheral protein or integral protein. Very quickly, peripheral proteins can be easily extracted, integral proteins cannot be extracted easily, and they need complete disruption of the membrane. This is important when you do some experiments, because you are not sure if the membrane is integral or peripheral, and then you have to use different techniques to extract them. Okay, wonderful. So this is the next slide that talks about a graphical representation of the different lipids which has been commonly found in a eukaryotic typical cell membrane. I don't know if it's very clearly readable, but I'm just trying to read you out from there that this ethanolamine, phosphatidylcholine, sphingomyelin, phosphatidylserine, ionositol, and so forth. Again, don't take too much pain in kind of remembering them, but at least if you remember a couple of them, it is going to suffice, like ethanolamine, phosphatidylcholine, choline, phosphatidylserine, and sphingomyelin. Just remember this first four. That is enough. We don't have to remember the rest of the items. But what is more important in this slide which I wanted to draw your attention is that if you see the distribution of the lipids in the two different lipid bilayers of the membrane. So this is the two different lipid bilayers of the membranes, inner and outer, right? Then you would see that vast majority of these lipids, if you see the lower panel, vast majority of the lipids are present in the inner leaflet of the membrane. Could you see over here? These of these are present as in the inner monolayer of the membrane. And these two lipids, phosphatidylcholine and sphingomyelin, are actually present predominantly in the outer leaflets of the membrane. So there is an asymmetric distribution of the lipids also. So we had seen that there is an asymmetric distribution of the ions. So in this entire class, I'm going to kind of use this word asymmetric distribution a lot of time. It will be annoying maybe, but that's important for us to understand this biological membrane. So bear with me. So we had first seen an asymmetric distribution of the ions of the different ions which is present within the cell and outside. Sodium ion, it is less inside of the cell, more outside. Potassium ion, opposite of that. Now we are talking about asymmetry of the membrane lipids also. Majority of the membrane lipids like phosphatidylserine, ionositols, 4-phosphate, 4,5-bisphosphate, phosphoric acid, all of these guys are present more in the inner leaflets. Only two of them, sphingomyelin and phosphatidylcholine, are more present in the outside of the membrane. So you could actually see over here that there is an asymmetric distribution of even the membrane lipids within the cells, okay, within the cell membrane. So that is again very important, and it is why it is important. I'm going to come possibly in this slide a little bit, that this asymmetric distribution is important. See over here what is written over there in the green. There is something called the phosphatidylserine. Now this phosphatidylserine ideally is present within the inner leaflet of the membrane. It has been found that when the cells undergo cell death, then the phosphatidylserine is been exposed to the outer membrane, or it is actually been exposed to the outside of the membrane. So there is a complete change in the distribution of the phosphatidylserine. When this happens, the cell usually goes for cell death, also known as apoptosis. We are going to learn about that more detail in the subsequent classes. And then we have these platelets. See, the platelets are able to play an important role. You know that the platelets plays a very crucial role in forming what you call as the platelet plug to the site of the injury, and that's how it prevents the blood flow and maintains homeostasis, right? That's what the platelets function is. The platelets act, for this to do, platelet has to first get activated. Normally platelets are inactive and they are not active. But once there is a tissue injury, the platelets gets activated and they start doing their function of forming a platelet plug. Now, in order to activate that platelet from its inactive stage or sleeping stage to an active stage, it's possible only when phosphatidylserine actually moves from the inner leaflet to the outer leaflets. So this asymmetric distribution of the lipids of the membrane is just not random. It is having some function, and many important functions. I've just listed two of them. There are actually at least 20 to 30 different functions why membrane lipids should be asymmetrically distributed. Okay, two of the examples are actually cited over here. Okay, now that I had covered a couple of slides, what I'm going to do is that I'm going to pause for some time, give you a little break to breathe in, and also for you to throw questions on me. So far, whatever you have understood, whether there is any problems, any queries, anything you want to add on, anything which is very sketchy and patchy and can make you clear, so feel free to ask to me right now. Okay, I'm just giving you a pause for 2 minutes. Anyone, any questions?
Sir, I wanted to ask, am I audible?
Yes, you are audible. Please ask.
Sir, I wanted to ask that in the peripheral membrane protein slide, it was written that for extraction we can also use alkaline pH. I understood why we could use high salt concentrations for their extraction because they would break up the electrostatic interactions, but how does alkaline pH aid in the removal? Please explain this one.
Which one? This high salts or alkaline pH? Alkaline pH
Yes,
means
sir. High salt is understandable, but alkaline pH.
See, alkaline pH means what? It's kind of mimicking a very mild detergent. If you take any detergent, whatever we use in the home, say Ariel detergents or whatever you use it, you measure the pH of that one. That will be little alkaline on the sides. So sodium hydroxide, I don't know if you are, if you could recollect, when we were kids, we kind of play prank in the classes, in chemistry classes. I mean, so what you do is that, there used to be soap over there, but then we always go and take a little bit of sodium hydroxide in the hand and rub it. So that gives a soapy feeling but the advantage is that it doesn't go as easily. So our professors used to call, hey guys, what are you going to do? So long you're washing your hands. But we say that, look, it's not going, because the sodium hydroxide is so much soapy in its nature that you keep on washing, rubbing, and your hands will be still very messy and alkaline. So what I mean to say is that, just apart from the fun, so that if you take an alkaline pH, it's something like a very mild detergent. That's what it's been said, that we don't need strong detergents. We want a mild alkaline condition, which is kind of mimicking mild detergent, as well as in salts, which is going to disrupt, as you rightly said, the electrostatic interactions. We could extract the peripheral membrane proteins. Did I answer your question?
Yes, sir. That answers my question.
Okay. So alkaline pH is kind of soap. Think about soap solutions, a mild soap. But when you talk about detergent, these are SDS kind of a detergent, sodium dodecyl sulfate. That's a very strong detergent. It's going to completely eat up the membrane.
Sir, good afternoon, sir.
Good afternoon.
Good afternoon, sir. I've got a doubt. Like, what's the difference between single and multiple transmembrane integral proteins?
Okay. Is there another question with you, or just only one question?
This, sir, only
Okay.
this.
So if you are going to go further detail about this different proteins over there, which is just beyond the scope of the class, because we have to cover different things. Many integral membrane proteins passes the membrane one times, like some ion channels. Many of the receptors are actually there, which kind of span the membrane so many times. Many of them I can just give you an example for your name, for just to remember the name. There's something called the G protein coupled receptors. We also call them as a serpentine receptor. Now this G protein coupled serpentine receptor, this spans the membrane 7 times. There is an enzyme which has been present on the membrane known as adenylate cyclase. What it does is that it condenses the ATP into cyclic AMP, and once cyclic AMP is actually been formed, then it activates something called cyclic AMP dependent protein kinase, and then the intracellular signaling cascade actually initiates from there. Now this guy, adenylate cyclase, is an 7 membrane spanning domain has there. So these are again membrane bound enzyme, but then it has been integrated into the membrane as if 7 times it has been passing in and out of the membrane. So that's the reason I said that there are membranes which spans once, there are sometimes membranes which spans twice, sometimes membrane spans multiple times, like the G protein coupled receptors and the serpentine receptors. And there is an additional flavor to this complexity, is that if you see these two sides of the membrane, there is a protein. Like these are all the integral membrane proteins. So protein will have an N terminal and a C terminal. N terminal is NH2 group, and the C terminal is carboxy group, COOH group. Many a times the membrane may be folded in a way that N terminal is outside, C terminal is inside. Another permutation, N terminal is inside, C terminal is not outside. Then there is for the multiple passes protein, it could have an N terminal outside and again C terminal outside, because it's going to fold in this way and then the C terminal comes out. Or you could have another combination where C terminal is inside and the N terminal is also inside. So we have this type 1, type 2, type 3 defined kind of proteins. There's a whole lot of biology in those kind of proteins, their functions, how they are incorporated within the membranes, and what is the relationship in respect to physiological different events. So there is a complete separate set of discussions. And we are not going to go over there, but again, if you want, we can actually talk some in a different forum. Did I answer your question?
Yes, sir. Too much. Like, yeah, I really understood. Thank
Okay.
you, sir.
Okay. Anyone else? Any other queries?
Excuse me, sir.
Yes, please.
Sir, the structural proteins in the membrane, are they are the proteins in the membrane, are they structural or functional or both?
No, they are both. So that's the fantastic thing about biology, is that everything is structure or functional. Let me put it in this way. Every single thing is structure or functional. And another important thing I should tell you, this is again forever important wisdom, is that any function has been dictated by the structure. Or in other words, if the structure is gone, the function collapses. So they are kind of goes hand in hand. So if you don't have a structure, you cannot retain a function. So that is the reason you take a protein and heat it. What happens is that it converts from its globular structure to its fibrous structure. The moment you do that, protein gets denatured, it loses its function. You heat the albumin, the egg. Every day morning we take an egg, right? We boil it. What you convert actually is that you convert a nicely globular albumin protein to a fibrous protein. and now the egg is rock solid. It's good for nothing. It's just, has to be broken down into and chopped into amino acids to be reused again. But you do not have any biological function out of that denatured albumin. So structure, function are all goes hand to hand. They are not separate. They are the two sides of the same coin. Did I answer your question?
Yes, sir.
Okay.
Sir, this I understood, but when we talk of like fibers like collagen fibers, we mainly say that they are structural and enzymes, they are functional proteins like that.
No. So that is a little superficial biology, as now you are in AIIMS and then you are going to further excel. Now you are now going to be an international standard. There is nothing in biology where everything is all and none. If collagen is, even though we say that it's structural function because it gives stiffness and other kind of things, there is a lot of functional aspect to collagen. So as we are going to study more and more, you will see that collagen offers a lot of places where there is an hydroxylations, phosphorylations that actually alters the collagen's functions, its interaction with other components. Every single thing you take it, any structural proteins, even if you take it, say for instance tubulins, the cytoskeleton proteins, actins, myosins, these are all structural proteins, but these are the proteins which have tremendous functional role. Without actin, myosin, you cannot have the spindle fibers, you cannot have the cell cycle, you cannot have cytokinesis, you cannot have cell divisions. So they are all together. At different level, we kind of teach the students a different amount of informations. We filter it out. We don't want to jumble yourselves with so much information, but as you slowly go up, and if your ambition for biology, biochemistry and all these kind of things are more, you would find that every single thing has this functional role. Structural proteins do also have a lot of functional role.
Yes, sir. That's a holistic idea. Thank you, sir. Thank you, sir.
Anyone, any other questions? If not, then I'll just take one question, then maybe I will proceed for the next, and again I'll give you a little pause. Yeah, please ask.
One question.
Yeah, please.
Sir, you are saying that phosphatidylserine when moves into the outer leaflet, then it means it is a marker of low-round cell death. But platelets start working when phosphatidylserine moves to outer leaflet. These are contradicting things.
No. So wonderful. So he asked that phosphatidylserine. Just now I say that just not saying, I had also displayed the slide, so I cannot run away from that reality what I told you. So phosphatidylserine normally is present in the inner leaflet. It goes and migrates to the outer leaflets. That's the signal for the cell death by apoptosis. Fair enough. But then I also said that platelets is able to play a role in the blood clotting, and that is possible only when phosphatidylserine moves to the outer leaflets. So logically, if you do in mathematics, this means when platelets gets activated, they should get killed, right? That's what logically you get an equation. But then the biological system are having a little more wisdom than just these simple equations. So whenever this kind of things happens, phosphatidylserine popping out, think about it. A cell has come into existence after 4.2 billion years of effort. So if we are seeing a cell in our body today, it has evolved 4 billion years back. Don't think that one single damn phosphatidylserine is going to decide the fate of the protein or the fate of the cell. Phosphatidylserine popping out is one of the factors which is seen in apoptosis or cell death, but it is never that phosphatidylserine alone is sufficient to drive the cell death. There needs so many other signals. A cell is not going to die. A cell has survived this Darwinian selection for the last millions of years. It is a very stubborn structure. It is not going to die just because one single protein is popping out. So phosphatidylserine is been found to be, yes, when there is an apoptosis, phosphatidylserine is been found to be out. But just because phosphatidylserine is coming out does not mean the cell is going to die. There are so many other ways to put the cell into check to prevent the cell death. So platelet activation is exactly the same thing. Do phosphatidylserine come out? Yes. Is it important for platelet activation? Absolutely. Does it mean that the platelets are getting killed when it comes out? No, because that is just one event. There will be n number of events which has to take place simultaneously for the cell death to be executed. So that's not completely true. Did I answer your question?
Yes, sir.
Okay. Okay, wonderful, good. So hold your questions for the next break. I'm just now proceeding for the next slides. Okay. Now this is the next slide, that shows you. So this is something which all of us are actually using in the winters. Glycerin soap, right? We use different glycerin soap in the body they kind of makes the skin smooth, and they are very, useful to use in the winter. So that glycerin is actually what you're using on the soap and gets drained in our toilet, is the one that is actually present on the cell membrane. It's very interesting, right? So the entire backbone is glycerin or glycerol, CH2OH, CHOH, CH2OH. That's the structure of the basic foundation of the phospholipids. Any phospholipids you are going to study in biology, it's a glycerin or glycerol backbone. That's what is actually shown over here. Now imagine, just because I don't have the blackboard and we have to just do a little bit of mental things to articulate the whole thing. CH2OH, right? CH2OH. So I put CH2O and that OH, that H I remove it and take another OH group from phosphoric acid. So now H and OH combines together to form water, and water gets kicked out. But this is an CH2OH is an alcohol, and then there is an phosphoric acid is an acid. What happens when alcohol combines with an acid? It forms an ester, right? So that's exactly what is happening. When glycerol actually comes in close contact to phosphoric acid, these two guys combine together, kicks out one molecule of water and get hooked together to form the polar head group, that is the phosphate group. So it becomes from CH2OH, it becomes CH2OP, right? First one. Now there is the next group over there is CHOH. Again that CHOH, it becomes CHO, and that H gets removed from this glycerol and OH gets removed from these fatty acids, and now it becomes CHO. Instead of now phosphoric acid, there is the fatty acids. Again another esterification of the carbon number 2, and the carbon number 3 happens the same thing like the carbon number 2. Again on fatty acid comes, water goes out, and again it forms CH2O fatty acids. So that's what exactly is the phospholipids. CH2OP, that's the polar head groups, because OP, P means phosphate. That's the polar head groups over here. Okay, this is the one which I'm showing over here, and this is the glycerol backbone. And these other two carbon atoms, the C2 and the C3, they have been attached with the hydrocarbon chains, so the fatty acids. These are the fatty acids that actually forms the membrane's lipid core, and this is the one that actually forms the polar head of the lipid. So the entire lipid structure is been based on the backbone of phospholipids. Phospholipids are actually been based on the backbones of glycerol. Glycerol is CH2OH, CHOH, and CH2OH. The CH2OH gets converted into CH2OP, that's the forms the polar head groups, and the other C2 and C3 participates by forming an esterification with the fatty acids, and they forms this long hydrocarbon chain. Hold on a minute, I showed one of the hydrocarbon chain as straight, the other one I had shown as with a little bend. I'm going to come that within a few seconds. But is this part clear to you all? I'm just asking you to unmute yourselves and give me the feedback, because this is where we are going to build up the foundation of this entire class.
Yes, sir.
Okay, you need to write down on your just on a piece of paper, and then it will be very easy. I mean, you have studied this esterification process. Okay, now over here what happens is that this is a wonderful thing that's happening. Hardly in any biological membrane you see that both of these fatty acid chains are aliphatic. This fatty acid chains in one of them also contains a carbon-carbon double bond. Mostly in almost all the biological membrane you find one of them is completely straight aliphatic, another one of them is also aliphatic, but it contains an unsaturation. Remember, these are all single carbon single bond. These are all sp3 hybridized carbons, right? When this is carbon-carbon double bond, this is an sp2 hybridized. And then if we have carbon-carbon triple bond, this is an sp hybridized. So this is an sp2 hybridization over here, and then we have a carbon-carbon double bond. And just because of that carbon-carbon double bond, there is a bend of about 120 degrees. That's in chemistry, no biology over here. But this chemistry facilitates the biology. Because of this carbon-carbon double bond, or because of this sp2 hybridization, this portion becomes what you call a small bend of 120 degrees. We use the word kink, K-I-N-K, kink. This kinking is very important. Think about it. If there had been no kinking, we would not have been living today in this world. That's the first fundamental driving force for forming and biological membranes. I'm going to come within the next slide that this kinking makes these biological systems inefficient. I'm going to talk about this inefficiency in a little while, but just remember that these double bonds gives an 120 degree bends, and now the structure looks like this. So one fatty acid chain is straight 180 degree, which is an sp3 hybridized one. Another one is an sp2 hybridized carbon-carbon double bond, which is bended to 120 degree. Okay, let me go to the next slide. Okay, this is something hydrophilic hydrophobic interactions. I'm not going to go into that. You guys know way more than what has been written in the slide. This is how this is known as a ball stick model. Just look, this is how the lipid bilayer actually looks like. The red colors are actually the polar heads, and the yellows are actually the fatty acyl chains over there, and the blue color ones are all the water molecules. So you could see that the water molecules are integrating with the polar heads, but there is no blue color water molecules with the aliphatic chains. Obviously, right? Water is a polar substance. The aliphatic chains are apolar, non-polar, hydrophobic. They are not going to mix together. But then again, there are water molecules from the cytoplasmic side, which is over here, again interacting and integrating with the polar heads. So these two polar heads, red color, and the aliphatic chain over there. That's exactly the ball stick model for a better understanding, ball stick model of this lipid bilayer in the membrane. Okay. Now the next thing what I'm talking about is a very interesting geometry over here. So it's a little bit of mathematics. We are not going to do any mathematics, but this is a fallout of a wonderful mathematics going on over here. Now imagine that glycerol has just one fatty acyl chain. I'm talking over here. In this case, you see that there is glycerol CH2H, CHOH and CH2OH, and then it has taken two fatty acyl chain. Imagine that it has taken just one fatty acyl chain over here. So now it has still a polar head, and it has an one fatty acyl chain. Now how this thing is going to pack in 3 dimension? This thing is going to pack in the 3 dimension in the form of a cone like structure like this, and we call this as a lipid maculae. Okay, but instead of this one acyl chain, if it has two fatty acyl chain with a kick or with a bend of 120 degrees, then it is not going to form this cone like structure. It is going to form a sheet like structure. Had it formed a cone like structure, it would have been perfect, no problem in that. But the problem is that once it forms the cone like structure, it cannot have any enclosure within it. It's going to jam pack it. It's going to pack very efficiently. It's going to pack too tight for anything to come inside. But once it forms a sheet like structures with two aliphatic chains, now it cannot form a cone like a triangular structures. Rather it will be like a sheet like structures. Now it forms a lipid bilayer. So the prerequisite for the lipid bilayer is that there should be two fatty acyl chains where it forms a double sheet like structures rather than a cone like structure. Okay, so this is something over here represented. Like if you see, this is how the membrane looks like. This is the two sheets, the outer sheet and the inner sheet, because of this reason that there are two fatty acyl chains. Now once you see this white spot in the sheet, this is these places where actually there are lipid aliphatic chains, right? These chains are actually represented over here. And now this can actually fold over here, right? And spontaneous is going to fold because these are apolar, and now the entire environment is actually polar. So it's going to kind of fold like this automatically. No one has to do anything. It's the geometry that is driving the entire reaction, and it's going to form like a seal environment. Now this is not like this, but this is like this. A sheet, imagine a sheet where two layers are actually lipid, a polar, and then intervening things is apolar. It's going, if you leave it as such, it is automatically going to fuse and form a spherical structure like that, where there is enough amount of space inside. That space inside is the cytoplasm, which we know today. So that is the first way in which the chemistry has given rise to this wonderful membrane and cellular structures, because of this presence of these two aliphatic chains. Now this is what I was actually tying, and then I'm going to pause in the next 2 slides. Think about this railway line. Like this is something like 2 straight lines, and this happens when both of these fatty acyl chains does not have any unsaturation. Now if they does not have any unsaturation, they are going to be a straight line like this. They are not going to have one with a 120 degree bend. Now it is something like this. Now if you take this one shown over here, then you will see that majority of these guys, the one which is straight like, if you try to fold them into a sheet like structures like this, it is going to fold and it is going to pack very efficiently, because these are straight lines, so perfectly aligned with each other, and therefore the entire structure will be absolutely perfectly sealed. What does that mean is that this means that the entire structure is fantastic because it is completely sealed and very nicely ordered. But what it means for the biology sense is that this structure does not have any fluidity because it is so jam packed that there is very little room for movement, because they had completely occupied every single space. Now if one of the fatty acyl chain has an unsaturation because of which there is a 120-degree bend, then they are not going to pack very efficiently, rather they are going to pack in an inefficient manner. It is this inefficiency because of this bending. It's something like, imagine that someone with a backpack has been going into a very crowded bus. What will happen? You will see that there is no person who can come close to you, because your bag is going to act as a buffer. It's not going to allow some persons to bump on you because the bag is going to protect you, right? This is exactly the same thing is happening. Because of this fatty acyl chain, what will happen is that there is a little bend, and because of this bend, two molecules cannot come very close proximal to each other. Or in other words, there is sufficient amount of space over here. That is what we call as the membrane fluidity. So the membrane fluidity is the outcome of an inappropriate, inefficient packing of the lipids on the membrane, on the surface of the membrane. Here we want the biological system to be inefficient, even though we always say that the system migrates towards, evolves towards, progresses towards efficiency. But here deliberately an inefficient system is allowed to survive because this inefficiency gives the membrane the required fluidity for the membrane function to take place. We don't want efficiency and we don't want a tight packing. We want a poor packing so that there is enough room for movement within the core of the membrane, within the plane of the membrane, and that is what is fluidity. This is actually shown over here in this particular diagram, that these are the different fatty acyl chain. You would see that the different fatty acyl chains has actually different charges, and this is what I was talking about. If you see the bilayer, see a lot of space, lot of gaps in between these two lipid polar heads. Because of this 120-degree breaks, and as a result of this 120-degree bends, they cannot come together, and then we have a lot of spaces. The other thing is very interesting over here, as I was talking about a little bit of like asymmetric distribution of the ions, then we talk about asymmetric distribution of the lipids. Now I am trying to bring the third asymmetry over here. There is an asymmetric distribution of the charges. Look at the charges. Majority of the lipids which is present in the inner membrane leaflets are all negative in charge. See, all of them is negative in charge. And majority of the lipids which is present in the outer membrane leaflets are either neutral or positive in charge. But surely the one which is present in the inner leaflets are all negative in charge. So there is also an unequal distribution of the charges in the biological membranes. So unequal distribution of the ions, unequal distribution of the proteins, unequal distribution of the lipids, and unequal or asymmetric distribution of the charges. Okay. Now I am going to show this slide. I am going to pause. This is the slide exactly what I was talking about, that the unsaturation. Now over here, as you know that in chemistry we have two kinds of unsaturations. We have the cis and trans unsaturations, right? The cis unsaturations are again highly like this over here in this one, right? These are the cis unsaturations, and these are the one which is trans unsaturations. Even though this one actually looks more symmetric in the same side, now the question is that once you have these cis unsaturations, we have more spaces over here if the two lipids are close by. Why? Because these are the two groups. There would be different functional groups over here, and there is something in chemistry called the steric hindrance. Because of this steric hindrance, these two groups cannot come close to each other. So imagine this is one molecule, there is another molecule next to that, and these two guys cannot come with each other because they are all oriented in the same directions. They are in the cis oriented. Cis oriented means they are all in the one side of the road. So if everything is in the one side of the roads, then you will have an steric hindrances, right? Because of the steric hindrances, this one is actually going to be very inefficiently or loosely packed. But think about this trans one. This trans one, the bulky groups are now present on the opposite sides of the road, and now there is always more chances of more efficiently packing this one as compared to this one. And that is the reason why the membrane lipids, the fatty acyl chains, are not only have an unsaturation, but I am going to add another point of that, is that they also have a cis unsaturation out of the trans, because the trans unsaturation is going to membrane again pack very tight, and again that is not desirable. Imagine that if you have suppose 100 elephants and you wanted to pile them together in the side of the road, if you put them all close to each other on one side of the road, they are going to bump on each other, right? But if you put one on either sides, then you are going to pack more number of elephants, right? Now the thing is exactly exactly the similar kind of things happening, that because of this bulky steric hindrances, they are going to leave out some space between them, and that is the origin of the membrane fluidity. So the origin of the membrane fluidity is the asymmetric distribution of the fatty acyl chains on the surface of the membrane, which are not only unsaturated, therefore giving a 120-degree bend, but at the same time these are also cis unsaturated, not the trans. Okay, now I'm going to start over there. This is what's exactly written over here. They are going to have this kind of a 120-degree bend, and this disorder causes greater fluidity in the membrane with the cis double bonds versus the saturated or trans fatty acids. Okay, now I'm going to stop here again, just 1-2 minutes break. I'm going to take up a couple of your questions. Anything you wanted to throw on me, please feel free to ask your questions before we actually proceed to the next sections. Please tell me. Yeah.
So why there is more negative charge inside the membrane, and there is almost neutral and positive charge outside the membrane?
That's what I said, that these are the distributions. This is how the lipids were actually distributed right from the origin in the membranes, that there is more distributions of these negative charges in the inside of the membrane rather than in the outside of the membrane. Now if you ask the second question, that why, the question of the why is something which I am going to tell you right now, because possibly this may not be very clearly explained in the textbooks. See that if you see the distribution of the sugar residues, what you call as the glycoproteins, where the sugar residues are attached, they are also asymmetrically distributed. There is more number of sugar residues on the outside of the membrane than in the inside of the membrane. Now if you see, majority of the sugars in biological systems are all sialic acids. Sialic acid is a 9-carbon containing amino acids, amine containing acids. It's not an amino acid, but amine containing acids, which is negative in charge. All acids are negative in charge, like COH or CO minus. Now think about it. If we have already something which is negative on the outer leaflet of the membrane in the predominant amount, can you have more lipids of the negative charge on the outside? No, it's going to have a negative repulsion. So it makes logical for the negative lipids to settle on the inner surface of the membrane. Now this is something, possibly they had this kind of an agreement right from the time of origin, that well, lipids, let me go inside with negative charge, and carbohydrates, you guys better be outside, because you have more power to do and recognition, cell to cell communication, so you stay outside, I'm going to stay inside. We cannot be on the same side because you are going to repel. Did I answer your question?
Yes, sir.
Okay.
Thank you, sir.
Anyone, any other questions? Feel free to ask any questions, even if it is related, unrelated. I mean, not too unrelated, obviously. Don't ask. Yeah. Any questions?
Sir, are all the integral membrane proteins totally buried? That is, they extend from the outer leaflet to the inner leaflet completely?
Majority of the integral membrane proteins are really dug into the membrane. Yes. That's what we call them as an integral membrane proteins.
Yeah, but sir, in higher secondary, we have read that some integral proteins are partially buried, some are totally buried.
That's what exactly what I say is that to the defined extent, if you see, they will be in the defined extent. There is nothing like all and none. So they will be, when while the peripheral membrane proteins are completely loosely held, but the integral membrane proteins, they actually go into the membrane to the different extent. If you ask me from the percentage range, it is anything from 20% to as much as 100%, and even more than that when it goes to the cytoplasmic side. So we have different integral membrane proteins integrated to the membrane to the different extent, but definitely they are within the lipid core.
Okay, sir.
Any other questions?
Sir,
Yes.
is there any subtle difference between the semi-permeable membrane and the selectively permeable membrane?
These are the terms which kind of we exchange and use it. Semi-permeable, selectively permeable. When you talk about semi-permeable, we talk more in the terms of chemistry. When we talk about selectively permeable, we now talk more in the terms of biology, because biological systems literally can actually see the things like, okay, I want to allow this thing, I want to allow, even if they are chemically chemically non-polar, but still, as I mentioned in the beginning of the class, just because something is non-polar does not mean that it is will have a free entry within the cell. So if you think about from the chemistry's point of view, if it is non-polar, it should go into the cell, right? But then biology comes a little more higher than that, because biology says that no, you may have been non-polarity, chemically you may be allowed, but I am not going to allow you, because biologically you are not important for the cell right now. So that then it comes as selectively permeable. So semi-permeable and selectively permeable, these are terms which we use interchangeably, but there is a little difference in terms of one more in the terms of chemistry and another more in the terms of biology.
Sir, is an egg membrane semi-permeable or selectively permeable?
Which one?
Egg membrane.
Egg membrane?
Yes.
Egg membranes, eggs are all biology, right? They are all biological systems. So almost all the biological membranes are selectively permeable. But when we say selectively permeable, they are also semi-permeable. But all semi-permeables are not selectively permeable. When you say selectively, means someone is selecting it. Someone is actively scanning the substances which is going to go inside the membrane or within the cell. Okay.
Yes.
Anyone has any questions on this packing of the membranes by the aliphatic chains?
Sir.
Yes. Yes. Kink is the kink. Kink means bending. Someone has asked. Yeah. Go ahead. Go ahead. Yeah.
Sir, is there any fixed proportion of saturated and unsaturated fatty acids in lipid?
Yes.
cell membrane?
Yes. That is something which possibly I'm going to come in the subsequent slides, but then you raised it, so you kind of prompted me to tell that one. So again, the proportion of this saturated and unsaturated fatty acid is different. Now, unsaturated fatty acid, as I just now mentioned, is the one that actually gives the membrane the fluidity. More cis unsaturated fatty acid, right? Now, if that is the case, now think about it. Suppose I am sitting in New Delhi here. The temperature is, it's winter, but still the temperature is, well, 12 degrees. Now, if I am at Chicago, United States, the temperature will be minus 12 degrees. If I am at a little more in the northern sides, like, you know, in Canada or somewhere else, the temperature will be maybe minus 20 degrees. Now, the same individual, the same Homo sapien has been inhabiting in all of these three places, right? Now, when they go to the colder environment, as colder it becomes, the membrane tends to become like a solid. The fluidity gets lost. And as the fluidity gets lost, the membranes gets overpopulated with saturated, unsaturated fatty acids with the bending, because it has to keep the membrane fluidity. Therefore, that is the reason why if you see all the animals, creatures, organisms which is surviving in an extremely cold environment in the Arctics, in the Siberia and other cold places, their membranes are more rich in unsaturated fatty acids. But if you see those individuals who are at a little warmer places, their membranes are rich. Their membranes do also have an unsaturated fatty acids, but proportionately less. Because the extent of the unsaturation, or another thing to be very clear to the entire class is that this entire composition of the membrane is nothing like, you know, something written on a hard ground. It is something which is very dynamic, very flexible. You migrate from one environment to another environment, your membrane's dynamic changes, your membrane composition changes, and it shifts to a different composition to suit the requirement. And the requirement is to keep the membrane fluidic. For that, if it needs that your membrane should be populated more with unsaturated fatty acids, so it will be like that only. So the proportions varies depending upon where you are located and what is the function that is needed out of that condition. Did I answer your question?
Yes, sir. Okay, got it, sir.
Okay. Okay. So let me go. So this is cis trans. Again, I'm going quick because we explained pretty, elaborately on this one. I'm not going to spend too much time. Now, this is something what is called as the membrane fluidity is temperature sensitive. That's what I was talking about a little while, right? So, you know, if you take the membrane, heat it, then it kinds the fluidity increases. It's something like, you know, take the butter, the Amul butter out of your freeze and then, you know, put it in a pan which is kept on a stove or an oven. It's going to melt, right? That's exactly what is happening. The membrane melts. Now, this membrane melting, if you see the dynamics of this one, this is kind of a sigmoidal nature, right? This is kind of a plot which you are going to study when you are going to study hemoglobin, oxygen binding to the hemoglobin. This is known as a sigmoidal kind of a plot. Now this plot shows that this temperature, as we increase the temperature, the membrane fluidity does not increase so much dramatically until we hit this magic point. This is what something called the melting temperature. This melting temperature is different for different membranes. It's not universal. Every membrane has their own melting temperature. But once you hit the melting temperature, immediately the membrane starts super fluidic and flows like a liquid. So this is how is the relationship between temperature and membrane fluidity. Temperature is going to increase the membrane fluidity. It's correct, but again it is not completely correct. If you are a part of the AIIMS community, you need to talk more than what the rest of the people talks in the different colleges. You need to say that, look, membrane fluidity is increased by temperature, fair enough, but then it does not increases continuously. There is an unequal increment of the membrane fluidity. It follows the sigmoidal nature of the graph. The temperature initially increases, the membrane fluidity gets it resists itself because the fluidity does not changes dramatically until we hit a threshold temperature of the melting temperature. Beyond the melting temperature, the membrane fluidity is going to dramatically increase. It's going to increase exponentially, and it becomes super fluidic. That's the relationship of the membrane fluidity with that temperature, not a linear one, but rather a sigmoidal one. You need to talk so that you explain the entire picture rather than just saying yes it's going to increase or no it's not going to increase. Okay, so this is known as the phase temperature, the melting temperature. Okay, now we are going to come next to a very important molecule of the membrane that is known as the cholesterol. This cholesterol, what it does is that it's known as the fluidity buffer. Very important. I'm just going to quickly cover, but this is extremely important, possibly one of the most important component of today's class. Just 2 slides, but very important. Cholesterol is the one that actually gives the membrane its membrane fluidity, but at the same time this cholesterol is also the one that makes the membrane less fluidic. How? So that's the reason we say that cholesterol is a fluidic buffer, or it maintains the buffer of the membrane. It acts as a fluidity buffer. Okay, now what happens is that this is how the cholesterol molecule gets integrated within the lipids. It fits within this, it intercalates within this, the lipid bilayer, and it sits over there. Okay, let it to sit. Then what it does next? Well, what it does next is that if suppose you take a membrane and you start heating that membrane, the membrane is slowly going to the transition temperatures and is going to increase the fluidity, right? Initially it's going to resist it because of the nature of the graph which I showed earlier, and then boom, it actually becomes super fluidic. Now within this phase, before it hits to the melting temperature, when you increase the temperature, then the cholesterol is going to resist the increase in the membrane fluidity. So if cholesterol is not been there, the melting temperature will be way less. Or in other words, cholesterol prevents the prevents the membrane fluidity and prevents it to become super fluidic if we keep on increasing temperature. So in a way, cholesterol is actually protecting the membrane because it is not allowing the membrane to flow like a liquid if you increase the temperature. Think about it. If you are going to Jaipur or Madhya Pradesh or some places in New Delhi in summers, in the month of June, the temperature will be almost like 50-51 degrees. Do you think your biological membrane is going to flow like a fluid? No, because cholesterol is where life-saving, that it comes and it prevents the membrane from becoming super fluidic and holds the membrane, and therefore prevents the membrane from flowing it. But now again you think that you're now going to Siberia, you're going to somewhere in Europe where the temperature is -10 degree. Do you think your membrane is going to be rock solid because entire thing is frozen? Something like if you take the coconut oil and put it in winter, you will see that it all becomes like a rock solid, right? Membrane does not become like that. Even though it has all the ingredients to be solidified, it still does not, because when the temperature reduces, then the cholesterol will try to increase the membrane fluidity. So cholesterol works in both the directions. When the temperature is increased, when the fluidity goes on increasing, cholesterol prevents it to become super fluidic. When the temperature reduces and the membrane fluidity reduces and the membrane tends to become like a rock solid, then the cholesterol comes into action and it increases the membrane fluidity. I'm going to show you one graph which is going to make you very easy for everything. This is the graph. If you are little, kind of favor of the graph, look at it. This is the blue line is with cholesterol and the red or the pink line is without the cholesterol. See, without the cholesterol, let's see the pink line. As you increase the temperature, the fluidity increases, not too much, because you have gone, temperature has increased from here to here, say almost like 40 degrees, 50 degrees, but the fluidity has not increased much, it is just almost here. But in this phase, slowly as you increase in the temperature, once it crosses the Tm, the melting temperature, you see how much the membrane fluidity is going to increase. If you have no cholesterol, the membrane is going to become super fluidic. Now suppose the membrane in the blue color with the cholesterol, see, when the temperature membrane without cholesterol is so much fluidic, but the membrane with cholesterol is this much less fluidic. That's the delta value. But at the temperature below the transition temperature, what happens is that cholesterol increases the fluidity, because now the blue line is more fluidic as compared to the pink line. So in order to summarize the whole thing in one line, you could say that below the transition temperature, cholesterol is going to increase the membrane fluidity. But above the transition temperature, cholesterol is going to reduce the membrane fluidity. And therefore, cholesterol is going to act as a very powerful calibrator, a buffer, a plasticizer, which is going to buffer the membrane fluidity so that the membrane does not abruptly changes between its extreme fluidity and still remains as a constant entity. Okay, again I'm going to pause for 1 minute and going to take up few of your questions, because this is a very important thing to what I had just now discussed. Any questions, again feel free to ask me.
Excuse me, sir.
Yes, please tell.
Hello.
Yes, go ahead.
How does cholesterol affects the fluidity?
Just now I mentioned you. If you want to know the chemistry for that, you have to read a little more, which you possibly may not be able to talk in this class. This is how it actually interacts with the different phospholipids within the membrane. But the net, what is the net effect? That's what I actually explained to you.
Okay, sir.
Okay, any other questions? I could not see the chat log, so please ask, unmute yourselves and ask, because the entire screen is actually in the slideshow, so I cannot see.
So,
Yes.
I want to ask, is there any relation between the melting temperature and percentage composition of the membrane, like percentage composition of saturated and unsaturated lipids?
Yeah, absolutely. I mean, what will be, that's what I said, that different membranes have the different melting temperature. Why it is? Because different membranes has different lipid compositions. So there is a very complex relationship between the composition of the membranes, the percentage of saturated, unsaturated, cis, trans fatty acids on the membrane surface, and its melting temperature. And therefore the melting temperature may be one temperature, maybe a particular membrane has a melting temperature, say for instance, say 40 degrees. That's an outcome of so many different things that has fed into the equation. So that's the ultimate one value, but that one value has been derived because of n number of molecules of saturated, n number of molecules of unsaturated, 2n number of molecules of cholesterols, 5n number of molecules of acetylamine of ethanolamines, this much amount of phosphatidylserine, and therefore it's finally the net outcome of those complex mixtures within the membrane that decides the melting temperature. Anything else?
Is melting temperature is directly proportional to saturated lipids?
So melting temperature, we will not be able to say that one, that melting temperature is directly proportionate to saturated, because as I mentioned that the membrane is just not one entity. The membrane has a lot of things within it. I mean, there are hundreds of different components. I just mentioned a couple of lipids. There are so many different lipids. So it is the outcome of everything. Which one is contributing to what extent? It's not very easy for us to tell piece by piece, unless until kind of do them in an in vitro experiments and then actually find out individually how much it is contributing. But all we are seeing is the net effect of something, what is actually happening, rather than individualistic contribution. So very hard. No one can actually tell that for this composition you are going to have this much. Many a times, if you logically, if you keep on increasing the unsaturated fatty acids, your membrane becomes more and more fluidic in its nature, right? That's the nature, that's the direct proportion relationship, that more the membrane contains unsaturated fatty acids, more easily the membrane will be fluidic in its nature. But then again, there are lots of kind of an ambiguity in that particular correlation, because many a times this is a biphasic response where you keep on increasing, then it actually drops down. So it's a complex thing. Anything else? Just to add a little flavor to this entire story, cancer cells has different membrane fluidity as compared to that of the normal cell. Therefore, people who studies biology from a physics point of view, that is known as the biophysics guys, they kind of study the membrane fluidity changes, and from that membrane fluidity changes, they can tell that okay, this is going to be cancerous, this is going to be non-cancerous. Not only that, they are also going to check the membrane fluidity over time and can predict that possibly this is going to be a pre-cancerous stage and is going to slowly going to be cancerous. So membrane fluidity has different applications. Many people use that as a metric to find out the changes, gross membrane fluidity changes between normal tissues and cancer tissues. And another piece of information, let me give it to you, that there is an Pfizer, there's a very big company known as Pfizer, right? All of you have heard of it. They are the one who is making one of the coronavirus vaccine. Now this Pfizer has something called the iBreast machine. What is the iBreast machine? That is a handheld machine that can be used in the mobile clinics, in the rural health setups, in the places where you don't have in tertiary healthcare like AIIMS or other places. There you could take that handheld machine, and that has been useful for breast examination for the females who are supposed to be in the high risk groups for having breast cancer. Now the whole philosophy of this iBreast machine is that they had an piezoelectric crystal in those ranges, and this sensitive, super sensitive piezoelectric crystal is going to pick up that, and it is going to tell immediately that okay, this is really not a normal breast tissue, this is possibly abnormal. Does not tell us for sure that this is cancer, but this is abnormal. That itself is a red flag. Then you quickly take that patient or that female and then actually go for the further testing, like mammography and other kind of an high-end analysis. But at least going to screen up. The whole philosophy of this test is change in the membrane fluidity, change in the elasticity, and that is going to be picked up by this iBreast machine, and it's going to tell you. So we have wonderful clinical outcomes of these observations. Any other questions?
Sir,
Yes, please.
is there any artificial product to maintain membrane fluidity?
Tell again. What you say just now?
Is there any artificial product to maintain membrane fluidity?
The artificial products to maintain fluidity is that if your food habit is actually different. That's the reason they say that don't take saturated fats, don't take transsaturated fats, right? Because if you take more and more of these fats, so people did those kind of fancy experiments where they kind of streamline the diet of this experimental mouse and experimental guinea pigs and fed them with a fat rich diet, with a saturated fat rich diet like ghee, like daldas, like vanaspatis, and then fed them with saturated fatty acids containing diets, and they found that initially, slowly over the generation, the membranes of those rabbits or those experimental animals has tremendously lost their fluidity, because now the diet is, the membrane is composed of the lipids what we eat, right? What they are not going to take it de novo from the air, so we get it from the food, and from the food the membrane actually gets the membrane lipids. Now if your diet is skewed to a particular diet regime, which is something like a fat rich diet, a trans fat rich diet, a saturated fat rich diet, this is artificial agents. Now we are changing the membrane fluidity. That's the reason why I say that apart from its atherogenic role, apart from its role in cardiovascular risk, the membrane fluidity is also one of the things that is even severely compromised if your dietary habit is not good. How you are going to get the results out of it? You see that people actually loses the sensation. The neuronal conduction is going to get affected, because membrane fluidity is a function of these neuronal conductions. Neuronal conduction is going to get affected. Ionic transport is going to get affected if the fluidity has been altered, and therefore the ionic potentials is going to change. That is going to change how good the neuronal electric conductions is going to be, and those individuals will have different kind of sensitivity. They will not have those finer movements, they will not feel the finer touches. Definitely it's not going to happen within a month or day, it's going to happen over the long period of time. But definitely that is the reason they say that have good healthy foods. What food you are going to take is going to decide what food is going to land up on your membrane. Did I answer your question? I couldn't. Your audio is not very clean. Can you just repeat it, ma'am, a little louder? Why cholesterol acts? That's what I was actually. Someone has asked this. I said that this is a very complex interaction of the cholesterol with the fatty acyl chain. Why is a question of chemistry? It's no longer biology, because cholesterol is cyclopentoparahydrophenanthrin ring. If you ask a chemist, they are going to say cholesterol is cyclopentoparahydrophenanthrin. That's a very complex, complicated structure with about 35-36 carbon residues. So that complex structure is going to interact with another complex fatty acyl chains in the lipid core. So there's a lot of complex chemistry goes on. We cannot say for sure exactly, neither it is important for us to be precise on that how cholesterol is working, but we know for sure that if cholesterol is there, then these are the outcomes of that one. That is itself is sufficient for us, rather than trying to find out, at least in this case, how precisely it has been doing. There is a lot of chemistry and physical chemistry over there. Did I answer your question? Okay. 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 Now I'm going to continue with the next one. I'm just lagging in the class because I have the membrane transport I have to cover. I don't know how I'm going to do it, so let's be a little fast. So so this class is until 4, right?
Yes, sir.
Yeah, hold on for
Yes, sir.
yeah. So I'll just request you,
Yes, sir.
if you don't have an appointment immediately after this, or if you don't have to go to a movie immediately after this, just hold on for 10-15 minutes. I'm going to finish up this entire block, so that you don't have to see my face for the next couple of months. Otherwise, I'll keep on bothering you. So in order to get rid of me, just hold on for 10 minutes after this 4 o'clock. Okay, so this is the one which I was talking a little back. Phosphatidylserine in the normal cell, it's inside in the membrane, and in an apoptotic cell or the cell which is kind of going to die, the phosphatidylserine actually goes out, pops out. Now that is something which is actually very interesting observations, and then phosphatidylserine kind of popping out is one of the signals. Again, this is not the only signal, one of the signal. Okay, that's what I was talking about. This is a wonderful term known as apoptosis, which is programmed cell death. Every cell in the body is been programmed to die at a particular point of time. So how this programmed death is executed, this is with the help of this kind of wonderful molecules like phosphatidylserine and of course other molecules. We are not going to go too much detail about this. I am going to also skip this slide because these are actually the experimental portions, where with the help of fancy experiments you could actually find out how much phosphatidylserine is actually been exposed to the outside of the membrane. 50%, 70%, 90%, 5%. So you need to do some sophisticated experiments, which we do in a biochemistry very routinely, to find out that how much is phosphatidylserine outside. Again, I'm going to skip this one. I'm not again going to talk. Now this is something very important I'm going to cover again. This is something, so far we are actually talking about membrane fluidity, right? I'm being actually using that term 100 times. Now the thing is that if one of you should ask me, how do you know that the membrane are fluidic in its nature? हमें कैसा पता चलेगा कि membrane fluidic है भी या नहीं? Why? How do you know to that for sure? Well, in order to know that for sure, you need to do a very brilliant experiment, but very simple. What the people did is that they took the membrane and then stained all the membrane lipids with the help of a red colored dye. What is that dye? Forget about it. That's not important. The important is that now the membrane looks like red blotch, right? Like this. Now what you do is that if you take this membrane, and then shine a laser on this membrane, ये membrane को आपने ले लिया, उसके ऊपर आपने एक laser beam से इस membrane को bleach किया. You bleached the membrane. Why laser? Why not any other light? Because laser is monochromatic. It's going to be very pinpoint. So आपने एक laser से इस membrane को एक छोटी सी जगह पे bleach किया. In other words, you have bleached the fluorescence light. So entire membrane is red in color because you have added a fluorescence dye. Now you have bleached that fluorescence light with the help of a laser, and now you are actually waiting in time that how long does it takes or what happens next. Slowly what happens, you could see that if you see in this one, you can see that the bleached portion is been getting repaired, right, slowly. And in the D portion you could see that your जो bleach हमने किया था जो step 1 में, that bleach पूरी तरह से हट गई यहाँ पे. The bleach is completely gone. What does that mean? That means that there is a movement which is taking place on the surface of the membrane, and that is the reason why this bleached portion is been kind of compensated by the migration of the lipids on the 2 dimensions of the membrane. Okay, my Hindi is horrible, trust me, because I'm a Bengali, so my Hindi is kind of horrible. If you score it in the scale of 1 to 10, it's possibly 11. So where 10 is the worst and 1 is the best. So mine will be 10.5 or 11. So I'll try to explain to you in English as much as possible, and Hindi will be very broken. So kind of excuse me for my Hindi. But what we could see that ये जो membrane की जो bleach किया हुआ है, ये bleach portion धीरे धीरे repair हो रहा है. And आप जब इस D में when you will see, आप completely देखोगे ये membrane पूरी तरह से recover हो गया है और हमें अभी bleach portion नहीं दिख रही है. इससे ये पता चलता है कि ये जो membrane का जो lipids है, that actually is migrating, और migration के लिए ये अपने आप में they are repairing it. That is the first proof that this membrane is fluidic in its nature and it has the translational movement of the lipids on the surface of the membrane. That is the reason why the membrane has been kind of repairing itself. Okay. Now again I am going to allow you to unmute yourselves and then come back to me. Is this technique is known as fluorescence recovery after photo bleaching. Fluorescence recovery after photo bleaching. If you follow the entire experiment, that is exactly what you did, right? We allowed it to bleach and then we allowed it to recover. So again, unmute yourselves. Any questions quickly? Just one or two questions very quickly. Any doubt in this experiment?
Sir, apoptosis, I do not understand that. The previous one.
Previous one is something which we has left in the time, so it will not be possible for me right now to go unless and until I cover everything and then come back to that one, because then
Okay, fine.
but I just kind of remind me if I am not covering that one, but feel free to remind me later. So this experiment portion is okay with all of you. Fluorescence recovery after photo bleaching. This is a short exam question that we definitely say that okay.
Sir, is there any unit for the experiment?
Is there any, what you say?
Unit for the measurement?
Yeah, if you see this graph, that's a good question which is already been kind of answered in the bottom of this graph. You could see over here that there is something called the normalized fluorescence intensity. So there is an intensity is something like the unit of the light. So with the help of this fluorescence intensity units, there is this arbitrary values are there, 0.1, 0.2, 0.4. You could see that there is a recovery. See, this is where we are done. This is the sharp drop in the fluorescence because of the reason that it has been bleached, and then slow recovery over there. So you could see that the luminescence units or the fluorescence unit is slowly going to get lost, and then eventually is going to come back to its stage. Okay, that is something what is actually been represented in this graph. Okay, I'm not going.
So this experiment only.
Yeah, tell again.
So this experiment only shows the lateral movements of the lipid, no other type of movement can be verified through this.
So when you say no other kind of movements, means what exactly and precisely do you want to indicate?
Like in high school, we learned the flip-flop movement.
Yeah.
Mainly flip-flop.
Now this experiment is pretty much on the translational movement of this in the two dimensions. So not the other kind of movements which is kind of showing over here. This is the flip-flop kind of movement. There are three groups of enzymes are there: flipases, flopases, and scramblases. Okay. What the flipases does is that it takes the lipids from the outer and brings to the inner. The flopases does the opposite thing. And the scramblases, actually what it does is that it randomizes everything. Inside to outside, outside to inside, doesn't matter. Now this flipases, coming back to your question, the flipases and the flopases activity, really we cannot do it, but we have different experiments to know that one. I mean, now we have hundreds and thousands of very sophisticated experiments like atomic force microscopy and then NMR spectroscopy and Raman spectroscopy and all these things, by which you could actually very minutely observe all those kind of different movements. But no, FRAP is only for the movement on the surface of the membrane. Now these are the three groups of enzymes: flipases, flopases, scramblases, which is going to take the substances from the outside to inside or inside to outside. Just think about it. I'm going to ask a very logical question. Think about it. Flipases and flopases and scramblases, which one is going to consume energy for moving or for taking this stuff inside and outside? Anyone?
So all the three would use energy, right?
There are both flipases and flopases, not scramblers.
Why? And why the three, all of the three will use? Anyone is giving any answer very quickly? What is the reason for that?
The flopases will take energy.
Or in other words, is this an uphill task? Is it a task which is energetically non-favorable? You are doing by investing energy?
So I think flipases will use energy, flopases will release energy, and scramblases will not consume energy.
Okay, wonderful. Okay, anyone has anyone wants to improve on this question? Okay, I had asked the question is that ये flipase and flopase, which one is going to consume energy? And scramblases, of course. इन तीनों में से किस में energy की investment ज़्यादा होगी? फ्लिपेज मूवमेंट में, फ्लॉपेज मूवमेंट में या स्क्रैम्बलेज में, कहाँ पे हमें एनर्जी ज़्यादा यूज़ करने की ज़रूरत होगी? एक्चुअली टू गिव यू द आंसर क्विकली, द फ्लिपेजेस एंड फ्लॉपेजेस दोनों में एनर्जी कंज्यूम होगा, but not in the scramblages, because think about it, in flipases and flopases we are actually moving through the lipid core, so there will be little change in the hydrophobicity, right? If it's a polar molecule sitting outside, it has to pass through the lipid core, right? Once it is passing through the core of the lipid, it is not going to be in a comfortable environment. It will not try to go into the lipid core. You need to literally push it through the lipid core, and therefore you have to invest energy. But scramblages does not, because here energy invested is going to be in a way compensated in a complex manner. I am not going to explain completely how it is, but this also involves the movement, but possibly it is an movement in an opposite direction, so somehow they kind of balances it out, and it does not actually needs an investment of the energy. I am going to put this.
There is that blue color thing which was represented there. Was it a lipid? I mean, is it a lipid core?
This is a lipid core, and this is a protein which has been embedded within the lipid, because this is not going to spontaneously go. It needs the help of some kind of a carrier substance driven process, right? If you take a lipid and if you allow this lipid to go by itself, the polar head even in 1000 years is not going to go, because energetically and thermodynamically this is not favorable, but you need an facilitator to do that one. And so these are the proteins which is actually, even then you need an investment of energy. It is shown in the second slide over here, flipases, flopases, you need to invest energies. And this kind of transporters are known as a P-type ATPases. So these flipases are P-type ATPases. When you say ATPases, this means they are using the energy of the ATP to drive this process. And this flopases are again kind of a protein that belongs to ABC transporters, again ATP binding cassettes. That's the full form of ABC, ATP binding cassette transporters. But the scramblages does not need an investment of the energy. But again, nothing on the earth is energy free, so that is a violation of the law of conservation of the energy. You could see over here there is a calcium ion over here which has been plugging in. Now when we have the calcium over here, this is a calcium driven process, and whenever we have the calcium over here to build up the calcium concentration, remember the intracellular concentration of the calcium is very less. Most of the calcium is stored within this calcium stores within the cell. Now in order to have mobilized calcium within the core of the cells, that is within the cytoplasm of the cell, you need to invest energy. And therefore, even though there is not an upfront investment of energy, but at the same time you are using energy to build up the calcium gradient, that actually uses energy, and eventually that energy is actually been used to drive this entire process. So flipases, flopases, both of them needs upfront investment of the energy, and the scramblages needs calcium ion driven process, which also has used energy at some point of time. So that's what is actually been represented in this particular. And flipases are P-type ATPases, flopases are ABC transporters, and scramblages are different. Okay, now I'm going to come, let me see. Okay, so I'm going to go to another important sections. Again, this is something which you might have to do a little bit of homework on yourselves, because again this is not something which you could do exhaustive way. This is something called anchoring the proteins to the biological membrane. So far you had seen that different proteins have been hooked to the biological membranes, right? किस तरह से, how it is. Now this is actually what you call as the anchoring of the membranes to the proteins. Remember that there are three types of anchors. This is known as the lipid anchors over here. Okay, these lipid anchors are there, which is helpful or which is important for anchoring the proteins to the membrane. Let me see if I have a slide. Okay, now there are mostly three types of anchoring. One is known as acetyl anchors, by which the proteins gets acetylated, and that's how they get anchored. There is something called prenylations, which is again another way of anchoring the proteins to the membrane. And there is something called the GPI anchors. GPI stands for glycosidyl phosphatidyl ionocetal anchors. So we have acetylated anchors, we have prenylated anchors, and we have GPI anchors. So GPI, prenylated, acetylated. These three anchors, just kind of remember this one by heart, because this is something which you frequently ask in the questions, that what are the different lipid anchors by which the proteins are actually been anchored to the membrane. Okay, these are the three different types of anchored. Now prenylation is there are couple of types of prenylations are there. This is known as farnesylations and geranylations. These are the types of prenylations. Prenylation is a big family. We have farnesylations and geranylations. These are the ways in which you can actually anchor the membrane. There is something which I am not showing over here, I am just skipping. And then there is something called the protein acetylation. This is another way of anchoring the proteins right to the membrane. I just mentioned there are two types of protein acetylation, that is known as myristylations and palmitoylations, depending upon whether there is a myristic acid which has been useful or palmitic acid which has been actually useful. Sorry, so someone has mentioned that I'm a little fast. I am indeed fast because I have to cover an entire set. So but then you had mentioned, so I'm actually now kind of scaled the gear down to third gear. Initially I was in the fourth gear, now I'm actually brought it back to the third gear, so a little slow, not too slow either, but little slow. Okay, so the proteins has to be anchored to the membrane. Because of this reason that there are three different kinds of lipid anchors: acetylated one, prenylated one, and GPI anchored. So what I will do is that I'm going to allow you to kind of ask your queries. Maybe once we discuss it, we'll kind of cover all of these things a little faster, and also come to know at least how much you are kind of absorbing it. So again, feel free to unmute yourselves and then ask your questions, whatever you have related to whatever we have discussed. We are almost end to this particular thing, and we are almost heading towards the end. So a few things are left over, but then this is something which you already discussed. So put your questions. So unmute yourselves and kind of ask your questions, and then as and when I am going to reply it, I am going to actually answer your questions. Yes, please.
Sir, am I audible?
Yes, please go ahead.
Sir, please explain the three anchors.
See, the anchors are actually the way by which you glue the proteins to the membrane. Okay, so there are three ways of gluing the proteins to the membrane. These are known as the protein acetylation methods. If you acetylate the protein, then you can anchor it to the membrane, number one. Number two is known as prenylation. If you prenylate a protein, then you can anchor the protein to the membrane. Now, prenylations are of two types. This is just a chemical name, prenyl groups, isoprenyl groups. There are two types of prenylations. One is known as the farnesylations, another is known as geranylations. So a protein can be prenylated as well as farnesylated. A protein can be prenylated and geranylated. So these are the types of prenylations. Then acetylations I already talked about. There are two types of acetylations, myristylations and palmitoylations. Again, these are the different ways in which you could hook the protein to the membrane. And then there are known as GPI anchored proteins, which is known as glycosidyl phosphatidyl inositol anchors, by which again you can take the proteins and hook it to the membrane. Did I answer your question?
Yes, sir. Yes, sir.
Any other queries?
Sir, could you please repeat the full form of GPI?
GPI stands for glycosidyl phosphatidyl inositol. It's a kind of a sugar anchor. The GPI one is actually mentioned over there in this box. So these are the different anchors. Why it's relevant? Is it relevant just for the exam? No, it's not. I'll tell you why. There is 40 to 50% of the human cancers are caused because of the activation of an proto-oncogene that is known as RAS. RAS. Now this RAS is a proto-oncogene that gets activated in 40% of the human cancers. You take the lung cancers, pancreatic cancers, stomach cancers, gallbladder cancers. You will have all of these cases where 40% of the individuals will have RAS activation. RAS is a proto-oncogene. If that guy gets activated, it's going to produce cancer producing signal within the cells. Now how is RAS? Where RAS is present? It is present on the membrane. How it has been anchored to the membrane? It has been anchored with the help of a GPI anchored. Now people are actually designing inhibitors by which they could notch out the RAS and drag it out from the membrane, so that RAS is no longer anchored to the membrane, and then the RAS mediated activation loop can be destroyed. So RAS activation is the cornerstone for many cancers, 40% of the cancers. RAS has to be activated because it has been bound to the membrane with the help of a GPI anchored. So any GPI anchored inhibitor is going to prevent RAS anchoring to the membrane, and thereby can cause the prevention of the cancers. Now does it make sense? Yes, right? Because now it is in clinical applications. So these anchors are very important.
Yes. Thank you, sir.
Any other questions?
Sir, could you tell something about the rate of diffusion of various substances through the membrane? Like
No, we had not reached,
nonpolar?
we had not arrived to that class. That's the class which is going to start possibly in 2 minutes. I'm done with the membrane structure function. I'm going to start with the membrane transport. That's the reason I told you that if any one of you are super busy, have some commitments, feel free to go. I will not hold you until beyond 4. But if you can stay 10 minutes beyond 4 p.m., then I'm going to cover this membrane transport. Okay?
Sir, in that FRAP experiment,
Uh-huh.
uh,
Yes.
sir, in that FRAP experiment,
Yes.
can we compare two membranes like the faster they take time in repairing, so the better the fluidity? Can we say that?
Yes, that's absolutely what we are doing actually in this case, is that we are also measuring this. See, if you remember that graph, can you can you see the graph on the x-axis of the graph? There is a time coordinate, right? That tells you that what is the time it takes to recover. Now, what you asked is exactly that the time of repairment, the time of recovery, the time to regain, the time to come back to the original conformation or original state is an index of the fluidity. If you see that has been altered, this means the fluidity is retained, but at the same time it takes way long to retain. It's something like a glucose tolerance test. There are some individuals who are completely normal, some individuals who are diabetic, but in between there is a very fuzzy group of individuals who are pre-diabetic, who are glucose intolerant but not grossly. They are in the borderline. The big problem is managing those individuals, because if you are little careful as a physician, you could still reverse them and make them non-diabetic. But if you are not good, then you are going to take those individuals and push them in the diabetic zone. So that individual's symptoms are pretty much very fuzzy, no clear cut. The blood sugars are very fuzzy, not normal completely, not completely abnormal. Now those individuals, if you give them a glucose load, then you will see that normal individuals can bring down the glucose load because they have the normal insulin secretions, and the glucose load is going to come back to the basal state maybe after 2 hours, 3 hours. But this group of individuals are also going to come back to the basal state, but not at 2 hours, maybe 4 hours, maybe 5 hours, maybe 40 minutes later than the normal cohorts. So that will tell you that these individuals are responding, but they are slow in time frame, and therefore they will have some predisposing problems. As of now, they may not be grossly abnormal, but they are going to land into diabetic zone sooner or later. So the time kinetics is very important. Time is just to tell you that whether you are going to be developing the symptoms later or not. So that's the reason why you have the time axis on the x coordinate. Does it answer your question? Okay.
Yes, sir, completely. Thank you, sir.
Any other queries? Okay. Okay, wonderful. So there are another 2 clinical conditions mentioned over here. The Tanger's disease. This is again because of the reason that there is a defect in ABC transport. As a result of this, the cholesterol and the phospholipids are not efficiently transferred. Okay. Again, this is a problem of these transporters across the membrane, and then you have this complete disease known as Tanger's disease. So there are a lot of conditions which is clinically associated with the membrane dysfunction. Okay. And that kind of covers me. But one thing, after everything, if you tell that okay, this is the membrane structure, we also know the membrane function. What is the name of this model? What model we know today is the known as the fluid mosaic model, right? So this is something which I should have told you earlier, but I deliberately kept it in the last slide, because you possibly has heard the name. And more important than this name is that once we have all this structure and wonderful beautiful thing hearing about the membrane, now it will be more easily kind of put into your memory that this is a double lipid membrane, and this is actually kind of the fluid mosaic model of the membrane. So if we ask in the question what is the fluid mosaic model, now you can explain it not only from the structural point of view but also from the functional point of view. Okay, so now I am kind of closing this one and going to the next one. Okay, let me see. Transport. Is the slide visible to you?
Yes, sir.
Yes, sir.
Yes, sir.
Just 10 seconds break before we kind of proceed to a, so this is very related. If you don't know the membrane structure, if you don't know the membrane function, then you will not be able to appreciate the membrane transport. So this are all goes hand in hand. But before we begin again with this one, with a new venture, any final question from anyone on the previous things? Okay, wonderful. No questions, miss. We are pretty okay to go, so let's proceed. So this is a membrane which, now I'm not going to talk about lipid bilayers and all these things. I understood that you already has understand that very nicely. Okay, now this is the one which is showing you the transport across the membrane. So this is gases, carbon dioxide, nitrogen, oxygen, small uncharged molecules, large uncharged molecules, ions, polar molecules, right? Now this has, all of this one actually goes through the membrane at the different rates. At the different rates, each one of them actually goes through the membrane. So membrane is pretty much permeable for these gases. Membrane is impermeable for these polar molecules like water, and definitely it is impermeable for glucose, fructose, the small ions, amino acids and so forth. Now let me ask you a question. That this carbon dioxide is way larger than potassium ion. What is the reason that this carbon dioxide is being allowed but the potassium is not?
So your audio was paused. We weren't able to listen to you
Is you can't hear me?
after you started. So now we can hear you after you started explaining the small uncharged polar molecules. The audio was totally gone.
Now can you hear me?
Yes, sir. Now we can hear you.
Okay. So what I asked the question is that this carbon dioxide is pretty large molecule as compared to that of the potassium ions and the calcium ions and the magnesium ions. Why is that these large molecules are allowed to go through the membrane, whereas the small guy like potassium ion, possibly which is one of the smallest ion in the periodic table, why this has been not allowed to go? Any question? Anyone can answer that question? One of you?
Hydrophilic.
Huh?
It moves by
Because
different.
carbon dioxide is non-polar and non-charged,
Okay.
so it can easily go through the lipid line.
And what about potassium ion?
The potassium ion is hydrophilic and charged,
Why?
so it will obviously.
Okay. Now what you are going to do is that, again, you are from AIIMS, so add a little bit of more flavor to this question. You need to tell, you have said, but you need to add a little more. That's brings an AIIMSonian. What is that? You need to say that potassium ion is being surrounded by a large hydration cage. So each of this potassium ion is been going to get surrounded by at least 4, 5, 6, 7, 8 water molecules. So this is known as the hydration shell or the hydration cage. And that in turn effectively increases the size of the potassium way larger than carbon dioxide. So it's not just the charge, it is also the final effective size of it. And that effective size is because of this high ionic nature of the potassium ions, the calcium ions, that they kind of carry with them. It's like VIPs. When the VIPs goes everywhere, you will see that there are 4 to 5 security guys, surrounding that person. The potassium ions are just like the VIPs and VVIPs. So they will be surrounded by a large hydration shell. Because of the polar in nature, that is going to make the potassium size huge than what potassium is actually, and that does not allow it to go through the membrane. Okay, wonderful, good. So yes, hydration enthalpy is more for small ions. Yes, when the small ions gets added with the water molecules, that stabilizes it, and once it stabilizes it, we release certain amount of energy from the system, and that is known as enthalpy for hydration. Enthalpy for hydrations actually are always exothermic in its nature, so they release a certain energy and stabilizes the system. Okay, now types of the membrane transport. We are going to come back that one hydration one a little. There is another slide which is over there, so I am going to kind of focus on that. Now we have different types of membrane transport: passive transport, active transport. Very gross. These two, big classifications. Passive transport means are of two types: simple diffusions and there is facilitated diffusions. And then the active transports are of two types: primary active transport and secondary active transport. Now what is simple diffusion? Anyone, what is simple diffusion is? So everything is actually been represented in this image. There is a simple diffusion, there is a facilitated diffusion, there is a carrier mediated diffusion, there is an active transport, there is a primary active transport, there is a secondary active transport. Now what is diffusion? Simple diffusion definition. Anyone? कोई भी बताओ.
So random movement of molecules which results net in movement of molecules from higher concentration to lower concentration.
Fantastic. Anyone else? Anything? Almost all of you are aware of that. I mean, all of you, if I ask every one of you, I'm pretty sure all of you know definition of the diffusion. So this is what is mentioned over here. The diffusion is the net passive movements of particles. It could be anything: ions, molecules, from a region of higher concentration to a region of lower concentration. Is that right? Is that anyone wants to add anything extra?
So it does not depend on any system.
It does not depend on.
The electrochemical gradient.
Yeah, that's what we have mentioned over here, that atoms, ions and molecules. So ions means there is going to be an electrochemical. So it could be a concentration gradient, it could be an electrical gradient, it could be a chemical gradient, it could be an electrochemical gradient.
If the electrochemical and concentration gradient are in opposite direction, then how will we define simple diffusion?
Opposite directions means? So the concentration gradient is going to go from a higher to lower concentration, and the electrical gradient is going to go in the directions where it is going to maintain the neutrality.
Yes. So if they are both in opposite direction, then
Does-
also simple diffusion can take.
Yeah, absolutely. Simple diffusion can take place. Doesn't matter. What is the one extra point what are you going to add over here? As an Amsonian, you need
It's independent of living system.
independent of living system. We are talking more in the living context. You are okay. You are right. Anything else?
No energy.
Yeah.
No energy is required.
Absolutely. When you say a passive movement, we kind of indicate that it's a low energy, it's a no energy investment process. You need to add one extra thing over here.
Slow process.
No, we cannot say it's a slow process always. It takes place, it's a function of temperature. So if the temperature is also there, then the diffusion is going to be faster. So what are going to add extra to this is that I'm going to redefine this. It does not need a membrane. Very true. That's the reason, it's a diffusion is the net passive movement of the molecules, membrane independent. We didn't mention anything about the membrane, right? Over there in the definition. So we are going to add, I'm going to read this definition again and say the diffusion is the net passive movement of the particles, atoms, ions or molecules from a region in which they are in higher concentration to the region of lower concentration till the concentration gradient vanishes. You need to add that extra point in that, because otherwise it is an open end. Is the diffusion takes place forever? No, it's going to take only till the balance is on the both sides. After that there is no net diffusion, right? There is the movement of molecules, but there is no net diffusion. So it's going to happen till the concentration gradient, till the electrical gradient, till the chemical gradient is being dissimilar or asymmetric. The moment it reaches same, there is no further diffusion. Do you agree with that?
Yes, sir.
Okay, wonderful. So let me quickly go. Let me go till the end of the slides, and then we can again take up the questions. Okay. So rate of the diffusion is proportional to the concentration gradient, hydrophobicity on the membrane, absolute temperature. Examples of the diffusions are so many blah 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 things. You are way aware of this one, so I am not going to talk too much about this one. So this is something which you have possibly done extensively in your high schools and exam prep. So this is the diffusions, and before diffusions, after diffusions. This is the classical image Again, I'm going to go. What is osmosis? Yeah, absolutely. I'm going to upload the slide. What is osmosis? What is the difference between
Diffusion
osmosis?
of water.
Huh? Tell again.
Diffusion of water.
Is there something which you need for osmosis? Yes, yes, that is very important. We need to mention that. So, movement of the solvent molecules. Again, movement of the solvent molecules through a semi-permeable membrane from its higher concentration of the solvent to its lower concentration until the concentration balances on both sides is what is the osmosis. And the amount of pressure needed to stop this osmosis is osmotic pressure, right? The counter pressure which you are going to put it to prevent the osmosis. Okay. Now there is something called the movement of the waters. Now water is a polar molecules. As I mentioned in the first slide, the movement of the water is something which is very interesting, at the same time it is very spooky. Why spooky? Because there are some places where water goes as it is, whereas there are some places where water is not been allowed to go. So that's the reason why I use the word very spooky, because it's kind of context specific. But where water is not been allowed to enter, like the renal tubules, there we have this group of molecules that is known as the water carriers. And what are those? Those are known as the aquaporins. So aquaporins are the water channels which helps the water to flow through the membrane, right? These are the water channels. Okay. All of you have heard of aquaporins before?
Yes, sir.
Excellent.
Yes, sir.
Okay, so transporters, I'm just going to summarize this. Transporters are of two types, channels and carriers. When you have channels, this is essentially an integral membrane proteins which is through and through the membrane, and then you have the carriers. That is the carrier mediated transport. It could be a primary active transport or it could be a secondary active transports, and again depending upon how many molecules are moving in which directions, you could have an uniport or you could be an symport or antiport, right? Most of these things you are pretty much aware of this one, right? Okay, so this is how the transport of these molecules taking place. This is what I was talking a little back, that potassium ions movement is through the water hydration shell. So think about this, like if you have a carrier and if you don't have a carrier, how does the thermodynamic kinetics looks like? If you do not have a carrier, then what happens is that if you take a polar molecule and literally make it to pass through the lipid bilayer, then this polar molecule, which is initially surrounded by the hydration shell, has to dehydrate, remove the hydration shell. This is an energetically non-favorable process, and therefore once passing through the membrane, it has to lose the stabilization of the enthalpy of the hydration, and that is the reason why the energy goes up tremendously. And then again when it comes back, again it gets the water molecules and the hydration actually goes down. So this is how is the kinetics. Now the kinetics of this entire things, once it has been actually aided by a transporter, you can see that the free energy involvement is way less than if they had been passing through the membrane just like that. So this clearly tells that the transport across the membrane with the help of a carrier is very favorable, because the investment of the energy is very less. We do not have to rip off the hydration shell, and the molecules can pass through the protein channels even with their hydration shells. Like when potassium goes through the potassium channel, it does have the hydration shell. So there is no question of getting dehydrated, and therefore there is no question of losing the enthalpy for hydration. So that is the reason why the proteins, the channel proteins and the transporters are so vital for the movement of the molecules. Types of the transport, this is an uniport, this is a symport, which is actually two molecules moving in the same directions. And you have these antiport systems. This is a part of the co-transport where they actually moves in opposite directions. These are all the different types of this transport. Okay, uniport, synport, antiports, or uniport and co-transport. Co-transports are of two types. Synports when the molecules are moving in the same direction, parallel trains, and then antiports when one is going from say Calcutta to Mumbai, another is coming from Mumbai to Calcutta. So that's the different directions, up and down train. So that's an antiport. Okay, wonderful, good. So we have this different kind of transport system within the cells. There is a chloride transport, there is a bicarbonate transport. There are so many of them. The basic principle is pretty much the same, that you create a gradient with the help of investment of energy, and then we utilize the gradient to move the molecules in. I'm going to come this one. This is already we discussed, so I'm going to skip this one, skip this one. Okay, this is the one which I am going to focus on this slide. If you could see the slide, this is a sodium glucose co-transport. Now, a very important process across the intestinal brush border epithelium. So you could see that there is a transporter system over there which takes into sodium and brings in one glucose ions, right? And this transport process is going to be something known as the sodium glucose importer system. And if you see what is the investment of energy over here, literally we are not investing any energy over here because there is a huge sodium gradient inside the cell, right? We are just seeing that 145 millimolar of sodium outside and literally just 4 millimolar inside. So this sodium is going to rush into the cell if given a chance. So sodium movement is in the direction of this concentration gradient, so it is going to rapidly move inside. Once it rapidly moves inside, it is going to also bring glucose within the cell. That is the sodium glucose transport, very crucial for maintaining the patients with dehydration with the help of ORS, which also contains glucose and sodium. If you just give the glucose, just sodium is not going to suffice. You need to give them both because this transporter actually has to bind with both of these two entities to be functional, and therefore the water loss from the intestine can be actually been compensated. You're going to study, and there is a lot of PBL also on that one. I'm not going to elaborate this, but this transporter across the intestine is very crucial. Now you might think that okay, this is a transporter that does not invest energy. Is that true? It is not completely true because sodium, to bring it inside, we had already invested the energy by creating a sodium gradient on the other side of the membrane, right? And this is with the help of the sodium potassium ATPases. Now this sodium potassium ATPases utilizes an ATP to throw the sodium from inside to outside, and this is where we actually invested the energy, and that energy is actually now been harnessed to flow the sodium favoring the concentration gradient. And therefore literally there is nothing like a free energy transport, but there is actually a redistribution of the energy. Investment of one energy in one front is been used to foster the movements of the molecules at the other end. Is it clear? Unmute yourselves and give me your answer in yes or no.
Yes, sir.
Okay.
Sir, the pump will also throw sodium into the lumen.
What you say? Tell again.
Sir, the sodium potassium pump will also throw back the sodium into the lumen. So
Yes.
40.
Yes. Yeah, exactly. So it is going to create the sodium gradient. That sodium gradient is actually important for it to absorb the sodium at the other end.
Okay, got it.
Okay, so now this is the summary. So we are kind of, let me see what is the time. So okay, so we are almost on the time, so I don't have any slides. So okay, so this is one summary, and let me just bring this one. There are, in addition to this transport mechanism which we talked about simple diffusions. I'm just kind of summarizing the whole thing. We talked about simple diffusion, we talked about the facilitated diffusion, we talked about the transport systems, we talked about symports, antiports, uniport, we talked about primary active transport and secondary active transport. Now we talked about all these different types of transport, but there is so many other ways cell can actually still communicate with the surrounding, and these are known as endocytosis, exocytosis, pinocytosis. I'm pretty sure all of you know that one. That endocytosis is what? That when cell engulfs large molecules, there is something called the receptor mediated endocytosis, where the entire receptor ligand complex is taken within the cell. And there is something called exocytosis, which is an opposite to the endocytosis, where secretions and the secretory granules degranulate and release. Like for instance, if a bee or some insect bites me, I will have a redness of that particular area, right? That redness is because of the reason that the mast cell actually degranulates and releases histamine, and that histamine is the one that creates that allergic reaction. Now this is possible because there is a degranulation. Histamines are contained within the histamine granules, but once they get stimulated, then they are going to degranulate and release the histamines, and that is the one that is actually causing the soreness and the redness in the site of the injury. Okay, so that is an opposite to the endocytosis, that is known as exocytosis. And there is something called the cell drinking, which is known as the pinocytosis, where the cell actually engulfs some fluid from the outside. So that is the transport. And in the other one, in the previous slides, we talked about the structure of the membrane, which is essentially lipid bilayer. There is integral membrane proteins, which is going pass through the membrane, and there is an peripheral membrane protein which has been loosely held to the membrane. Again, when you say pass and pass to the membrane, different proteins are being integrated to the membranes to the different extent, ranging from all the way to 10% to 100%. Again, we had also seen the important role of this fatty acyl chains and the unsaturation in kind of taking the membrane fluidity. If the fatty acyl chains are unsaturated, the membrane fluidity is more. If they are cis unsaturated more than the trans, then the membrane fluidity is even more. And we have also seen the role of the cholesterol in producing a membrane fluidity buffer environment, because if the membrane is been too solidified at a low temperature, cholesterol increases the fluidity, and if the membrane is too fluidic because of a high temperature, then cholesterol reduces the membrane fluidity, and that is how the cholesterol acts as a buffer for the membrane fluidity. And the last we had seen that how the proteins are actually been anchored to the membrane with the help of the different protein anchors. There are three types of them. The acetyl anchors, which involves again two subtypes, the myristoylated and the palmitoylated. Then we have seen the GPA anchored proteins, which is glycosyl phosphatidyl and acetyl anchors, which is the way in which the RAS protein is being hooked to the membrane. And we have also seen the prenylated anchors, which are of two types, the geranylated and the farnesylated. Now all these geranyl groups, farnesyl groups, you will be amazed to know that these have been derived from cholesterol biosynthesis. So think about it, cholesterol biosynthesis is taking place at one corner of the cell, but the products of this cholesterol metabolisms and the cholesterol biosynthesis produces these farnesyl groups, then these geranyl groups, and that is the one that has been utilized to hook the proteins to the membrane. So the cholesterol metabolism is so fantastic that it provides the raw material for the substances to be hooked to the membrane. And then this membrane anchorage is very important because they had a lot of relationship to the different human health and disease. So that kind of finishes my block of the membrane things. So if you have any queries, any questions, any doubts in this class, in the previous class, subcellular organelles, please feel free to ask me right now. I'm going to be here for the next 5 minutes. And if you want to discuss anything related to this one, or to other things which has been related to biology related to this stuff, feel free to ask me for the next 5 minutes. I'll be happy to take your questions.
So can you explain the graph of bleaching experiment?
I didn't get your question. Can you repeat it?
Can you explain graph of bleaching experiment, why that was a very low peak?
Which one? Which one?
The bleaching experiment.
The flap one?
The red colored
Flap one? Okay. Let us go back to that one. Hold on. What is the query?
So why there is a low peak at just before 100? There is a very low
Yeah.
fluorescence intensity.
Fluorescence intensity, we have destroyed it with the help of this laser, right? हमने लेज़र शाइन किया है इसी कारण ताकि वहाँ पे जो फ्लोरेसेंस मॉलिक्यूल्स है ना उनको हम ब्लीच कर सके, ताकि वो फ्लोरेसेंस मॉलिक्यूल वहाँ पे मर जाए. And then this fluorescence molecules, if they have been gone, now we are asking this question, okay, now I have destroyed the fluorescence molecules. How much time it takes for the fluorescence molecules to repopulate, to come back? Now I will ask one question to you. How do you know that this is because of the translational movement of the lipids on the surface of the membrane, or because of the de novo synthesis of the lipids from inside the cell? See, cell is also continuously synthesizing lipids. So how do you know that lipids has actually moved in and filled the space, or is it that new molecules has actually came and they had filled the space? Is the question clear to you?
Yes, sir. The process is very quick. So de novo is should not be a possibility, because within a few seconds only a good amount of fluorescence is reached, which should have been possible or we.
Let me take.
Sir, I think
Let me take this to.
fluorescence is less than the original one. That's why.
Sir,
Tell
when
it.
you check the fluorescence of the adjacent space, they will also have decreased a bit.
Not so
Sir,
much.
initially fluorescence was exactly 1, but after this thing, the final fluorescence is less than 1.
Yes. So had it been not de novo lipid which has been not think about it again, this is not going to be a very clear cut answer, because if there is a de novo lipid synthesis, those lipids will not be fluorescently labeled. See, जो लिपिड सेल के मेम्ब्रेन के ऊपर पहले से हुआ था, पहले से था, उसी लिपिड्स को हमने फ्लोरोसेंस डाई टैग किया हुआ है और उसी लिपिड्स ही हैज पिक्ड अप द फ्लोरोसेंस इंटेंसिटी, राइट. अभी जो सेल्स के अंदर नया जो लिपिड्स बन रही है, those are not labeled, so even if they come and fill up that space, they will be patches or dots of the black dots only within the black dots, isn't it? Because they are not labeled. We had labeled only those in the initial population, we didn't label them later. So in order to kind of piece together this particular problem, what we do is that we kind of add in addition to this one an inhibitor for the lipid synthesis. See, now we are going into the experimental biology. So initially I told it in a little superficial way. So what we additionally do is that we also prevent lipid biosynthesis, so that or the lipid transport, so that we don't allow fresh lipid molecules, fresh phospholipids to come and populate this area. We want completely in isolations. So you synthesize जो lipids जो cell के अंदर बन रहा है उसको आप stop कर दो थोड़ी देर तक, because this experiment can be done within a couple of minutes only, and then you completely squeeze that internal lipid synthesis process so that no internal lipids can disturb this, and then you see that whether the regain is there or not. You will see regain, but definitely this is see the panel A, this is way brighter than the panel D, right? That it is brighter. Why? Because the panel D में ना already the lipids are redistributed, so the intensity per square inch is little less because they had redistributed. Any other questions?
Sir, how is cat report different from a channel?
Tell again.
Sir, carrier with a unibot, how is it different from a channel?
So, okay, so when we say carrier, so there is something called the ping pong mechanisms. Possibly you may have heard of it. If not, we, I'm telling you. So channels, when we say channels, these are integral membrane protein all the way through and through the membrane. It is something like a tunnel which someone has actually dug for smuggling some goods. So that is all the way through it. But what happens is that the carriers, mostly the carriers, they diffuse through the membrane. यानी carriers क्या करता है, जो suppose glucose carrier, GLUT, right? And let me see if I have the image over here. I can pull out that image and show you, if at all I have the image. So the glucose transporter somewhere. So what the glucose
transport
in the oh, you are right. I am looking at a different slide. Yeah.
Previous lower one.
I wanted to. Okay, wonderful. Yes, great. You are right. So this is the one. Okay, the glucose transport. What it does is that actually this is actually showing a different transport, but the GLUT transport. I wanted to show you the one, the GLUT 1, the GLUT 2, GLUT 3, GLUT 4. The glucose transporters. What they do is that they operate by a mechanism that is known as ping pong mechanism, just very similar to that of the enzyme kinetics. So they will have one conformation. That conformation is going to bind to the glucose. Once it gets bound to the glucose, it is going to change the conformation, and now it is going to start diffusing through the membrane, carrying with it the cargo, which is the glucose, and it goes to the other side of the membrane in the cytoplasm and again changes the conformation and releases the glucose. So it is something like a missile is a particular rocket launcher is carrying a missile head. So it's been just transporting the glucose by actually diffusing through the membrane. That is the reason why we say that if the membrane fluidity has been compromised, the channel proteins or the movement of the ions through the channel may not be compromised, but the movement of the small molecules like this will be compromised, because this involves diffusion through the membrane, and that is going to be dependent on the temperature, and therefore the transporters and the channels are different in this way, that channels are always through and through, but this transporters actually goes and passes through the membrane, That is how they operate in a slightly different fashion. This, did, is it is it is it ans- did it answer your question?
Yes, sir, very fine. Thank you,
Okay.
sir.
Anything other than this? Any other questions? Even questions from the previous class? Anything? Okay.
Sir,
Yes. Go ahead.
sir, after cells die, the membrane becomes semi-permeable or impermeable. What does it?
When the cell dies, then its capacity to retain the membrane semi-permeability is gone. Now the cell actually becomes just like a passive container where anything can go in and anything can go out. That's the reason why I had mentioned that philosophically the entire life is actually maintaining asymmetry, fighting against the nature. Because what the nature wants to do is that it tries to maintain the symmetry, and because the symmetric stage is the most random stage, and because that's where everything has been very equally distributed between all the sides. But once we are actually a biological membrane, a living cell, it tries to maintain an asymmetry. We had seen the asymmetry at the different fronts, right? Lipid asymmetry hai, and then charge asymmetry.
Yes, sir.
So this asymmetry is important. If you lose the asymmetry, that's the first point for going towards a cell death. So, and when the cell completely dies, then it becomes completely symmetric. This means the outer sodium concentration, inner sodium concentration same, potassium concentration same. What will happen when the outer and the inner sodium concentration remains same? Is there a potential difference? No. In the absence of any potential difference, you will not have action potentials. So the cell is actually as good as a dead cell.
So, sir, in boiled eggs, the egg membrane is now completely permeable.
In the boiled egg, the egg membrane, it is now a function of how much did you boil it. So if you kind of boil it sufficiently, some people actually take a little softy boiled egg, some people take a very hard boiled egg. Some people likes that yolk to be a little liquidic and fluidic, so they boil it little less. If you boil, now that you have boiled it, you have denatured the protein function is gone. The semi permeability and all these things are actually out of the window. Now if you boil it too hard, the membrane will have large pores, so anything can actually enter through it. It is now something like a big mesh kind of thing. But if you boil it a little less and take the egg in a little raw form, then it will have a smaller holes, and then this many things will enter, may not enter. So now it is out of the purview of the biology, because now you have actually kind of converted the globular protein to a fibrous protein. You convert a native protein to an protein which is denatured. So that is a different kind of a thing.
Okay, sir. Thank you. And please upload these PPTs to Server platform.
Yeah, and again for the entire class, whosoever is here and whosoever has left, please communicate to them that if you have any further questions, just email me, WhatsApp me, because I don't know when you are going to meet again. So feel free to kind of ask your questions, email me, WhatsApp me, SMS me, just clarify your doubt if you have anything related to this one, and we can always discuss this and settle it out. So once you have the PPT, possibly you will have more doubts, so feel free to ask me, and we can always sort out on those questions. Okay, so with that, so
What's the number?
so can you note down my number? So whosoever is noting down, just feel free to note it down. 9999. There are 4 times 9, 612564.
4 times 9, 612564.
Perfect. That's what exactly is. And my email is also pretty straightforward, even though my name is very complicated. So it's very confusing. But I mean, just Google my name. You would have something popping out in my name. So just write Supradeep Karmakar. You would have my lab web page coming up, and you will have my email address over there. So you can always write me, or I can WhatsApp me. I can send you my email. So with that, we kind of take and leave. Stay safe, stay healthy, and then enjoy your days. If last few days at home before you kind of pack your bags and come to AIIMS, leaving your parents. So this is one of the finest day. I mean, thanks to COVID, this is some of the finest day where you could actually spend a little more time with your beloved one. So enjoy those last few days. Take it easy. Biology is always there. We can always study all these things. The whole life is there, but this is a very precious moment. and then spend with your loved and the dear ones, and then when you are going to come back, then we will again meet. So have a safe day. Bye bye.
Thank you, sir. Thank you, sir.
Thank you.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir. Thank you.
Thank you, sir.
अच्छा उनको पीपीटी ठीक है, मैं बाद में भेज देता हूँ। ठीक है, मैं